asking a question. So why I'm very happy, because the audience here, the teachers are from different backgrounds, and some of you, my young students who are sitting in the crowd, they also are from a different background but having a common theme of hope, that is a biology. My lab focuses on infectious disease, so I am a medical microbiology by training. Throughout my tenure in the career, I worked with infectious disease, trying to understand that how such a tiny bacteria or a virus can actually make us ill. With few thousands of gene to 30,000 gene in the body, what that we do not have, that we are still not able to answer a simple question of how to take care of infectious disease. The entire world is crying with antimicrobial resistance. You open a page of anything, be it CDC, WHO, newspapers, everywhere the cry of antimicrobial resistance, death is increasing, so etc. etc. So today I'll take you through an interesting story of a bacteria, and that is how the title is Life in a Vacuole. So the pathogen of the choice is a Salmonella. Sanjay, this pointer is, where is the pointer? LED, it will not work, right? Wait, now it's okay. So just, thank you so much. That's fine. Yeah, thanks a lot. So the pathogen of the choice is Salmonella. Just for the crowd who is not much aware of the bacteria, this particular pathogen, Salmonella, is a pathogen which affects the body through orofecal route of infection. That means if you take contaminated food and water, and if the bacteria is present there, it will get into the gut. It will, in fact, be able to take care of the acidic pH of the stomach and then land up in the small intestine. Now, pH of the stomach is roughly around 2 to 2.5. Anything that is there should be actually chewed up. But these bugs are so smart that they have their signaling mechanism to bypass that acidic pH and then finally end up in something called as the small intestine. So in the small intestinal lining, if you still remember your 9th class biology, the small intestinal lining is not naked. It is lined by very thick mucus, which is around 150 micron thick mucus. And on top of the mucus lies your gut flora. What we call it, as a second brain, a very big group of research on the so-called gut microbiota or the microbiome. I'm sure that you all must have heard of it. So this microbiome protects us. But if that is the case, how these Salmonella can enter? That was a question that was a mind-boggling. You have a thick mucus, on top of that you have a microflora, 230 million different species. It's like a mat. So how can some 100 bacteria just come and enter our system? System is so well guarded, but it happens. We suffer from systemic typhoid fever, we suffer from gastroenteritis and diarrhea. So what they do is, the nature is sometimes very interesting. Though you have a very thick the thick mucus lining, some of these cells, like dendritic cells, like a M cell, they throw their arm in such a way that they rise above the mucus and above the flora. And these bacteria, they beautifully come and slip inside. It is like just sliding and slipping inside the hole. And that is how their systemic intervention happens. So they get inside the system, and they can be transcytosed from the apical side to the basal side, and from there they disseminate into various different body organs. That is how this bacteria causes the disease. Now, why Salmonella of all? According to the WHO, this had been put as a high priority pathogen. Till a couple of years back, this was in bioterror category number 2. But due to the devastating effect on the morbidity and mortality, this has been put in bioterror category number 1. So this is a pathogen of concern. Anybody who is not very careful with the food and water, please try to become very careful, because you do not want this bug to get inside your system. It is a debilitating disease. It's just not some kind of a fever and you get cured. There are long-term implications of someone who actually gets this bacteria inside the body. Now, what happens when it gets inside and it gets disseminated? It resides in this cell type. Now, this cell is known as a macrophage. Macrophage is a cell in a body which should clear anything that gets inside. It should take up and it should get destroyed. But what they do, these bacteria, they will not allow them, but on top of that, the bacteria gets inside and they rearrange their entire structure of the macrophage and act them as a vehicle to go from one site to the other. And all this is done, we will discuss about it, is done by a certain gene in the bacteria, which we call it as a pathogenicity island. These are the big chunks of DNA which is acquired by horizontal gene transfer into the bacteria, which gives them a fitness advantage. And I call them as an amazing secretion system injection supramolecular needles. They use this needle structure to deliver the effector protein into the cell to take it up and have their life. There is another pathogenicity island, and this was my own work when I was a postdoc, that how this particular pathogenicity island will allow them to replicate. Now, why do I call it as a life inside the vacuole? I'll come to that. Now, Salmonella is an intracellular bacteria. What is meant by intracellular bacteria? Suppose a bacteria gets inside the body, but it does not want to enter any cells. You call these bacteria as an extracellular bacteria. They will be floating around in the body. They may get cleared by the blood cells, or they may actually just float and go elsewhere, and eventually that can cause sepsis, or the patient will get rid of that. But intracellular bacteria are like Trojan horses. If you have read the story of Trojan horses, that how very sneakingly the bacteria gets inside the cell. Not only that they get inside the cell, suppose now this is an example, this is a beautiful experiment done by my colleague Sandeep Ishwarappa. In fact, he is the first PhD colleague with whom I started my lab when I joined IISc in 2004. Sandeep was a medical student, and he was my first PhD student also. And he very patiently used to infect the cells. Currently, of course, he is an associate professor in the department where Professor Ganesh is, that is in biochemistry. And he was someone who had such a patience to look under the microscope to figure out that where this bacteria actually resides. You see here, this is a cell, macrophages, which is labeled with an antibody to RAP7. Now, RAP7 is one of the protein which can mark the Salmonella containing vacuole. And why do we call it as a vacuole? Because each Salmonella, if you see here, the green is a GFP expressing Salmonella. Each one of them, they stay in a single compartment. Not only that they stay in that single compartment, which is bound with a boundary, they never leave that compartment. Now, can you imagine very stringent life of the Salmonella? It is in a very tightly bound compartment here. This green bacteria is staying in a tightly bound compartment. This is just one of the marker for the Salmonella containing compartment. There are 40 proteins from the host and some from the bacteria which makes this house. And they don't leave the house. Then how do they survive? It's not that it is very spacious compartment and then they get enough food, etc. On top of that, the pH of this compartment is acidic. It is not even a neutral pH that they can survive very well. So stringent pH, low nutrition, absolutely no way that it is very luxurious. Then how do they survive and how do they become so successful? So what was figured out, so you look at this EM structure, what it shows is this is indeed in a vacuole. This is a Salmonella, just look at this. This is a rod-shaped bacteria, by the way. Salmonella is a rod-shaped gram-negative bacteria. And it has a peritrichous flagella. That means it is a flagella all over so that it can move. And of course, it's a gram-negative, it does not form spores. So when you see there, see this is a Salmonella. Of course, it depends. It's appearing to you as a round because the electron microscopic section was cut in that particular plane. So it is bound in a vacuole, and what is very striking about it is the bacteria, once it enters, will it only stay there? No, right? There's a right to live is the right to reproduce. So it will reproduce. Now you know about the bacterial division. So it's a binary fission. 1 bacteria into 2, 2 into 4, it's a logarithmic growth. So in that case, if this bacteria has to divide, what is the status of the vacuole? Will it come out of the vacuole, divide and then go again? So that was not the case. In fact, the Salmonella divides along with its own vacuole. So the bacteria divides and the Salmonella containing vacuole also divide, which make them to come in a single Salmonella in a vacuole. And this was something very interesting. So how bacteria divide along with the vacuole was a question that is still puzzling us. It's 2 decades in the lab. Some wonderful work has been done, and not only that, this particular bacteria is an example of the first example of something with such a tight fitting vacuole. You do have like Legionella. They live in a big spacious vacuole, big home. This is the one which is a very stringent vacuole. And you know, if it comes into the cytosol, the life will be much simpler. But no, they don't want that. They stay within the vacuole. The question is, how do they divide along with the vacuole? That is the question. So the first thing is that how many bacteria does the Salmonella containing vacuole harbor? That was a main interest. If it is a big vacuole containing multiple Salmonella, then what happens is the bacterial number goes down and the host wins. That means if it's a big vacuole with many bacteria inside, the Salmonella dies and the host wins. But if it is a single bacterium per vacuole, then the host dies and the bacteria survive. That means that the single bacterium per vacuole or the vacuolar lifestyle is a strategy for the bacteria to establish successful pathogenesis. Now, bacterial fission is okay. So there is a lot of beautiful mathematical dynamics that plays a role here. I mean, we have done a beautiful modeling to understand that what is the capacity of a cell to harbor how many number of SCVs. First question. What is the threshold that the bacteria can undergo how many generations to have the cell? Is it something that when it is filled up, will the cell burst and can it then reinfect the population? And if it does it come along with the vacuole? So the secondary infection will be the bacteria along with that compartment coming out and infecting. Then what will be the dynamics? The dynamics may be something much more interesting than a naked bacteria which is coming from environment. So all of them extremely interesting questions that kept us really busy. But we scratched our head, and how do you take this very surprising fact that Salmonella containing vacuole is dividing? Because vacuole is made up of host protein, all the host protein. What initiates the cues for the division? So we took some very good, a cue from mitochondria. As you know that mitochondria is a good example of an endosymbiont, and it is none other than an alpha proteobacteria which once upon a time had harbored the cell and it became one of the important organelle, and mitochondria also divide. So DRP1 apparatus, this is one protein which is important in making a site of mitochondrial division site to give rise to 2 mitochondria. So we asked a question, does the Salmonella containing vacuole also uses the same DRP1 machinery as it uses in a mitochondria to make this bacteria to get into 2 vacuole? And indeed yes, partly yes, because when you treat the cells with the SOV, sodium orthovanadate at a particular concentration is going to inhibit the DRP1. So if you do that, what we see is the multiple bacteria per vacuole increases, and with the multiple bacteria per vacuole increasing, the intracellular bacteria or the life of the bacteria within the cell goes down. That means the Salmonella containing vacuole must be a single bacterium per vacuole, and if that is perturbed, then the bacteria will meet with a dead end. Now, my chemist friends here, you will be the best one to actually ask a very important question. It's a multi-billion-dollar question, that can we find a small molecule inhibitor which is going to inhibit the vacuolar division? You don't worry about the bacterial division. If the vacuolar division is perturbed, the Salmonella cannot survive. How much ever number big bacteria, bacterial growth you have. The bacteria will not survive if they come in a single multiple bacteria in a vacuole. And indeed, it is true if you also prove it in another way. Say for example, if you use P50 dimethyl, which kind of quenches the DRP1, you see that all the bacteria, they come in a one vacuole. So you have again a multiple bacterium per vacuole because your DRP1 action is somehow inhibited. This is a control where you see that yes, there is a single bacterium per vacuole. When you have this multiple bacterium per vacuole in the DRP1 condition, and even in the INT4 cells, which are the epithelial cells, you see this. See this particular part, multiple bacteria in a vacuole. And all of these bacteria, in fact, when you do their live and dead assay, they die. So they are not able to establish the infection. So we have a really amazing tool now to understand that don't tackle the bacteria per se, because it's a right to live. If we have a right to live, so are they. And if you perturb their life, that is what we are facing, the antibiotic resistance. So don't perturb that, but you think intelligently about how do you perturb something else, so that you can at least manage the infection status. And we again figured out that this Salmonella is very unique in perturbing this cell functionality. Wild type means it is a bacteria which is infecting the cell, and the moment the bacteria gets inside the cell, all the lysosomes, which are supposed to be the end fate organelles to take care of your exogenous life forms, to clear them off, all these so-called volume of acidic compartment which you see in the uninfected cells are completely gone in the infected cells. It can mean two things. First, the bacteria may secrete a factor which may neutralize the pH of the lysosomes, or it may somehow modify the expression pattern of the lysosome forming gene, that no lysosomes are being formed. So what is it? Of course, by again very tedious experiments we figured out Salmonella does not neutralize the pH, but it sequesters all the cargo which is going to make the lysosome toward making its own Salmonella containing vacuole. So they make lot of SCVs and the lysosome number suffers. So there are not enough lysosomes to actually take care. This is a controlled concanomycin. Concanomycin is a VATPase inhibitor, and it is going to actually neutralize the volume of acidic compartment. The bacteria acts actually exactly like that of the VATPase. So to that extent it neutralizes the volume of acidic compartment. And indeed that is not only in the cell line. If you take a mouse which is infected with if you take a mouse which is infected with Salmonella and you harvest the spleen is an organ where bacteria goes. Yeah, there is something called as a splenomegaly. The spleen becomes enlarged. You can take the spleen and you can just it's a soft tissue, so you can just tease it with two glass slides, and then you sort out the cell and you see that wherever there is enough bacteria, the pH of that particular compartment drops. So it is indeed true that the volume of acidic compartment is significantly dropped by this infection, and this may be one of the very intelligent strategy by the Salmonella to establish its growth within an organ or within a tissue. So one more intervention strategy can be thought about. How do you not allow the lysosomal cargo machinery to fall? So you have to constantly upregulate the machinery so that the lysosomes can be formed to take care of the infection. And this would be a good intervention strategy. Now, to prove further that you need to really do, we came across something which is interesting. It's just not the mitochondria. Nowadays, as a cell biologist or as a chemist or as a physicist or mathematician, we are always constantly challenged by one factor, that how do you really prove that that is the case? It's so much in biology. It's just not a one-way contact. Now, if it's just not a mitochondria, because mitochondria makes a contact with the endoplasmic reticulum, and then it has some other contact with the lysosome. So now we are talking about a three-way contact. Here also, so it's just not the mitochondria we saw, but when you have the KDL sequence which can mark the ER also marks the site of vacuolar division. So mitochondria and ER both are playing a role in marking the site of division. Now what puzzled us for a very long time is, we can understand that a bacteria divides, it's a binary fission. 2 micron bacteria undergoes to give two daughter cells of 1 micron each, fine. But then what orchestrates the division of the vacuole because vacuole and bacterial division should orchestrate with each other. And they should really coincide to give rise to two bacteria in their individual vacuole. How does that happen? That orchestration is important. And after very tedious experiments, we figure out that even the ER comes and wraps around this Salmonella containing vacuole to give it a site to give it to two bacteria per vacuole. So yeah, there are a lot of these beautiful snapshots of the construct where you can see that, if you can see here, that this is a dividing bacteria and you see a constriction here, and this particular constriction is the one where this actually the ER is getting wrapped. We have some very amazing live imaging microscopy also to see the same. I'll just skip this for a moment. And these are all the additional experiments to prove that you have to do some dominant negative DRP1 and ask the question that what is important for the growth of the bacteria, for the division of the vacuole. And every single experiment proved that single bacterial per vacuole is required. It is a key for the growth. Now, as I mentioned to you that the intracellular lifestyle is very important, and you see here very clearly that this is a growing bacteria, this is the vacuole, and when the bacterial constriction happens, the vacuolar constriction also happens, which leads to two bacteria in a vacuole. Okay? Now this is quite clear, and we know that, and otherwise you see them all in the single bacterium per vacuole. You will never see in a cell multiple bacterium per vacuole until and unless you use some intervention strategy. So this is very clear. Of course, for mitochondria, it is an amazing endosymbiont, and we know that even if you talk about a lot of these cells as a organelle formation, then you work into the dynamics and you see that when the mitochondrial divide, how the division happens within the mitochondria. Now, coming to the ER, as I mentioned that ER forms a very important point of contact. The ER stress is important to make the ER to change their status from a sheet to tubule. So ER actually also exists in many other forms, like it can be as a sheet or it can be as a tubule. So the Salmonella infection indeed lead to the increase in the ER stress. I'll show you how do we see, and why did we focus on ER much more? Because we realized that the moment you have a growing Salmonella, you need more of the membranes. Otherwise, from where are you going to draw the membranes for? And why the ER should be giving up the membranes? You know, so the ER should have a long tubule with which it should wrap around like a rope, which is wrapping around any object. And it is indeed true that a couple of the ER stress factors like ATF4, they are all increased upon Salmonella infection. Don't worry about this particular mutant. So in our lab, because we have a battery of mutant with all the pathogenicity factors, we generate the mutant in the lab. Now this particular mutant, SSAV, cannot secrete any of the spike effector proteins. So they are kind of attenuated strain. So whatever the wild type is doing, this particular mutant is not doing that, because they get killed, they get cleared by the cells. They are a very good control for us. And yes, it is indeed true that there is a stress involved. Then what you do is you go on counting, it's a tedious experiment again, the number of cells, and you figure out that all these proteins, be it ATF4 or ATF6, upon infection over different period of time, the stress response is high. That is for sure that the stress response is high. Sure, with another protein that is the ATF6, again the number of counting shows that the stress response is very high. But then how does it implicate? What is the implication of this ER stress with what I was talking about is the division of the vacuole. What implication it has or what connection it has. Now, Salmonella infection, as I mentioned, it leads to the structural changes in the ER. And what are these structural changes? Now, this is a very beautiful schematic which shows that how there is a connection between the nucleus and the ER. These are all the proteins which makes these connections and the channels, and we concentrated. So this part was carried out by my, my very recent young colleague who graduated from the lab, Umesh. Reticulin 4A, that is one of the very important connections that is there to make these point of contacts. And it is indeed true that all these transcript level of be it reticulin 4A or CLIM63, specifically reticulin 4A and CLIM63, these protein increases in the wild type, not in the mutant, not in the only uninfected cells, but in the wild type. That has a significance. Why should it increase? I mean, why should reticulin 4A increase when the cells are getting infected? And we figured out that yes, it is indeed increased. So again, you have this KDL sequence, and you see that the KDL sequence is exactly marking the site of the dividing bacteria. So the percentage of the tubular ER increases dramatically. Sheet is of no use. So the sheet conversion to the tubules. Now we are trying to understand that actually how many tubules, what are the dimensions of the tubules, and how many turns will it happen. So we have a very good collaboration with our physicists and the mathematician across. And in fact, they only gave a hint that don't think that because the bacteria is dividing, there is a suction force which will allow the vacuole to suck in. That is not the case. There is something which is happening due to the live bacterial processes. So you must define what are those live processes. It is not as mundane as a suction force which is giving you per bacteria as a vacuole. And that is how in fact we could do so many beautiful experiments. So yes, it's a tubular ER, and the tubular ER always increases in only infected cells, not in the bystander cells. The bystander cells means in a given plate you will have some number of bacterial which gets inside, but the other cells will be uninfected. Those are called as the bystander cells. Why do we have to do experiment with the bystander cells? Because there can be something like a signaling which is produced by the infected cell which can affect your bystander cells. And one has to always, if you are working with two life forms, a cell, a bacteria, a virus, parasite, always make sure that your system will not have 100% infection. So you should look very carefully into the pockets where in a plate you have an infected cell surrounded by non-infected cells. So what happens in those non-infected cells? And yes, the bystander cells in fact have no effect. It is just like the UI. So whatever the sheet to tube conversion, tubular conversion, is only there in the infected cells. So saying that it is definitely required for the expansion or for the wrapping around. And how does it play a role in the SCB division? That is the question. Yes, then you do experiments with all these, the constructs which is going to be inside the cell, and then you look into the fold proliferation of the bacteria, which is a hallmark of a life. Indeed, when you transfect your cells with the reticulin 4A construct, look at the fold replication. They go very high. This is an empty vector, the normal fold of replication. Moment you have a tubular reticulin, the fold proliferation goes high, showing that the marking is extremely important. And when you knock it down, how important is reticulin? So you knock it down by using the shRNA, and you see that really the fold replication drastically decreases, showing that yes, it is very true that it helps in the SCB proliferation. Again, huge number of very tedious time-lapse imaging to show that the reticulin 4A is very important, or 661 is very important for the vacuolar division. So I will just see whether I can play this video. If not, this is one beautiful video which shows. Okay, so this video shows, if you really focus on this particular, this was one bacteria, and you see here that how now this is getting wrapped, and then you have the two bacteria. So this live imaging in fact clearly shows that endoplasmic reticulum forms a point of contact and it wraps around, which leads to two bacteria in a given vacuole. So the bacteria divides along with its vacuole, and this is what is an amazing finding that we got. And finally, again, did lot of other time-lapse imaging to show the same, that at what time point, look at, these are all the lapses, time lapses, and a different frame shot to show that indeed this bacterium gets marked by the endoplasmic reticulum tubules, and then they get into two bacteria. This all convincingly proves the same. Again, a huge number of experiments, other kind of softwares to show that how is a groove, how the wrapping is done, etc., etc. And we were not very successful in doing the film-SEM because the instrument was not there in India. And we are trying to now, collaborate with Australian group and indeed do this film-SEM experiment to prove. But other than that, you see a very clean marking or the imagination and the very clean division along the axis of the bacterial division and the vacuolar division. So yeah, so far so good. ERT views facilitate the SAB division, and if you compromise that, the bacteria comes in one vacuole and the bacteria dies. And there is no doubt in that. But then the question comes, what makes the ER comes into the site of the division? So till so far we know that the ER tubules marks the site, and if you have the less in the ER sheet, there is a less SCB inside the cell. So the tubules make the SCR sheet grow like this. It's beautifully marking the site of the bacterial division. But what marks the site? This was $1 million question which was bothering us because it cannot be only from the host site. There has to be something from the bacterial effector protein which is attracting or recruiting the reticulin foray onto the site of the division. And we were lucky, in fact, to figure or stumble upon an effector protein which is called STEA. STEA is an effector protein or a virulence protein of Salmonella, which in fact is essential for the SCB fission. If you do not have STEA, so when we deleted the STEA gene, you see the number of bacteria per vacuole increases. So multiple bacteria in a given vacuole. Not only that, when you have a multiple bacteria, look at the STEA mutant, all in a big blob. And this blob could not survive because they get attacked by lysosomes very easily. It's a bulky vacuole, no? So they get targeted to the lysosome easily and they get completely cleared off. So we realized that STEA is the one which marks the site and which is important for the division. And so now we have an amazing model showing that it is the STEA, but the question still remains how the STEA brings the endoplasmic reticulum. That is still remaining. We could not figure out that what is the dynamics. STEA comes, it marks, and then the reticulin is wrapped around like a zipper model. Or it is the some other protein which is tethering the STEA with the endoplasmic reticulum. So there are still a couple of more experiments which are required to be done. But without any doubt, if you have a mutant of STEA, they are not able to proliferate. And as you see here, the single bacterium per vacuole in the wild type where everything is intact and where the STEA is knocked out, it's a multiple bacteria that comes within the vacuole. So, and the SCV diameter also increases. We are now trying to understand the purifying these SCVs, which is again not a very easy task, to purify these bulky SCVs, and then you take this, analyze these individual SCVs in terms of its dimension and the content and the life inside. So let us see how much we will be able to do. I am going to just tell you that because they are now in a single big vacuole, bulky vacuole, they are very easily targeted by the autophagy machinery, and they are somehow getting cleared off by the lysosome, by the other autophagy machinery, by the very sheer of it's able to not decrease the lysosomal number. So the wild type, it is able to actually decrease the lysosomal number, but when you have this STEA, the lysosomal numbers are high and they target the, the large vacuoles. So it is indeed true that STM wild type can decrease the lysosomal number, but the STEA cannot. In a nutshell, if I really mark the summary here, it is very important for this particular bacteria, Salmonella, and I have no other examples of this bacteria. This is a very unique one, to live in a single bacterium per vacuole and never leaving its vacuole. They are quite happy, so the life in the vacuole is the key for their successful pathogenesis. How do they do that? They not only get inside the vacuole, but they have a very successful lifestyle by dividing its vacuole. Sorry, just going out 1 minute. Yeah. So they have a very successful lifestyle by dividing its vacuole, and if they are perturbed in their division by either not having a STEA or not having the reticulin tubules from the host, they end up in this bulky vacuole, and these bulky vacuoles are not able to divide and grow, and that brings down the pathogenicity of a bacteria. So if we have to really think of a very smart intervention strategy to counter this disease of typhoid and gastroenteritis, we cannot discover more antibiotic. It's of no use because it will make resistant. What we need to target is how do you inhibit the vacuolar division, that is one, or how do you somehow inhibit the wrapping of the reticulin around the Salmonella containing vacuole. So these two can be an amazing strategies to counter this infection. I'm very blessed always with my group of colleagues, very vibrant colleagues, all in red in this column. They have all graduated and they are faculties or postdocs. These are the ones who are currently in the lab doing their PhD. So that is how I'm here. They are extremely vibrant. And because I am a microbiologist by training and I totally believe on the right to live, it's not that my somehow my passion and love for microbes. So this is what I wrote, a very small piece. This is a piece which I wrote for tale of antibiotics. This is one tale. Very sorry, it's going on and I don't know how this is working. 1 minute. This is a very small piece that I wrote for all of you. We are tiny, we are small. We need microscope to see us all. You need microscope to see us all. Still, we are mighty and will kill you all. Give us stress, give us pain. Still, your efforts will go in vain. We are community, strong and high. Treat us with respect and we treat you right. That's what is happening because of the antibiotic stress. We are not bad, but you made us so. We will become worse if you continue to do so. Hold back and keep quiet, and we will be harmony throughout our life. This is actually the harmonious conditions that we are all dreaming for, and hopefully we will do that in your future. Thank you very much, and open to questions.
Thank you, Professor Dilshika, for an excellent talk, and then open for some couple of questions.
Yeah.
So good afternoon, ma'am. I'm Srijit, and I do have 4 questions.
Yes, sure.
My first question is, apart from the STEA, are there any other possible factors that affects the that helps in the proliferation process?
In the proliferation of bacteria inside. So good question. From our pull down, we could not actually find anything other than the STEA, which can make all the bacteria come within the vacuole in a bulky vacuole. So there may be some other factors which can aid in the vacuolar division. Proliferation, it will have, see, STEA mutant, they don't have any role in the in vitro or in the environment, they grow. But the moment they are within the cell, these bacteria are attenuated because they cannot divide their vacuole.
My second question is, how does STEA helps in lysosomal trafficking?
Okay, STEA in general has nothing to do with the lysosomal trafficking. Only what it is doing is it is allowing the bacteria to somehow recruit the endoplasmic reticulum tubules to give 2 bacteria in 2 vacuole. Now the vacuolar structure or the cargo is made from the similar protein by which the lysosomes are made. LAMP1, LAMP2, others. So all the cargo is getting sequestered toward the growing number of SCV. That is how you are getting a low number of the lysosome. That is how indirectly STEA is playing a role, because it is giving more single bacterium per vacuole.
So there is no movement of lysosome towards the periphery region during infection?
No, the number of acidic volume or the number of lysosome itself is completely down. So there are no lysosomes. It looks as if you have treated the cell with a VATPase inhibitor, concanaminase.
My third question is, how does ER know where to wrap around the construction?
Very good question. That is what was the entire. Now, how does ER know where to wrap around? When we looked very carefully, we realized that it is the ER tubules which is exactly wrapping around where the bacterial fission is happening. Now, the question is, how does it orchestrate? The small invagination which is created, now that invagination is because of the mitochondrial wrapping around of DRP1? Is it a tripartite contact with STEA? That we are trying to understand, and we have not solved this mystery yet.
And my final question is, what signals the ER towards the vacuolar central and allows it to wrap?
What? It is all in the vicinity. If you really see a very enclosed, compartment, the tubules are longer. Now, why the tubules are longer? Because it induces the so-called ER stress. So you do see a sheet to tubule conversion. But it is always like a serendipity that why they are always in close positioning with the Salmonella containing vacuole, right? So yes, it is all there, and that is how the STEA makes them to wrap around.
Okay,
Yeah.
thank you.
Thank you so much.
Any more questions from anyone? Okay, so
Yeah.
is there
One question.
okay, there is one. Okay.
It was an interesting talk, ma'am. And I read one of your papers in which you mentioned sirtuins
Yeah.
and its connection with Salmonella and its pathogenesis.
Yeah.
And I have read that sirtuins are also a part of hypertension. So is there a possibility that some of the protein has one part which is mechanically activated, which has some parts in the endoplasmic reticulum also, which makes it mechanistically sense the fission which happens in the Salmonella, so that it senses and wraps around nicely.
Good thought. See, sirtuins are histone deacetylase, and normally it is not a part of hypertension, but it has a pleiotropic, lot of pleiotropic effect. Yeah, they play a role in diabetes, hypertension, so-called ventricular atrophy, etc., etc. So their knockout has a pleiotropic effect. Now, sirtuins also play a very global role in the metabolic flux of fatty acid to the glucogenesis, or the glucose uptake and their further conversions. Sirtuins play that role. I have never thought from that angle, that how it can play a role in the vacuolar division. But we know that from our experiments with sirtuins, that it play a role by perturbing the metabolic flux. What it does, if the host flux is towards the glycolysis, it reduces the fatty acid oxidation in the bacteria and vice versa. But yes, it's a good thought process. I'll think over it. Thank you.
Okay, so with this, again join me. Thank you, Professor Deepshika, for an excellent talk. And now, so it's my pleasure to invite the next speaker. So he's Professor Kumar Swami from School of Chemistry, University of Hyderabad. Welcome, sir.
Thank you.
Ya, welcome, sir.
Good afternoon, everybody. Our Nirmala auntie told us that it is the hand of God which determines many things. I was supposed to talk first, and fortunately for me, I was delayed by Indigo flight. And thanks to Chakravarti, she has taken over the first this one talk. Okay. Now, whatever I am going to talk to you today is not completely about my research or any such thing. Okay, it's a popular talk so that students and teachers will be motivated towards doing some chemistry at least. That is the reason why I have chosen this particular topic. Only the last slide shows something about what research I am going to have been doing, and I am going to do in future if there is if there are enough facilities. Okay, the topic that I have chosen is serendipity in chemistry, Prussian blue and ferrocene. Okay, with inputs on coordination and organometallic chemistry. Okay, the main compounds that I am going to talk about are this one. Okay, that is Prussian blue and ferrocene. Okay. Alright, in the periodic table you know where iron comes. Okay, this is another way in which you can represent the periodic table. I am not going to the details of that one, but then we'll straight away go to I am going to tell you what about the things about serendipity as such. The word, okay, was coined by some Walpole, and the origin is the Arabic name of for Sri Lanka. So Serendip means land of lions. Okay. Now what happened long back was that three princes of Serendip, okay, it's a fairy tale sent by their father to explore the world. They were always making discoveries by accidents and by sagacity of things they were not impressed of. Okay. But then when they were going along, when they were just walking, a camel driver stopped them on the road and asked if they had accidentally seen one of his large camels. Now, the way in which they were trying to answer this question is like this. Those princes asked him, was the animal blind on his one eye? Was it lame in his one leg? Was he loaded with honey on one side and with oil on the other? These are the questions which they asked. That's how they probe and they find out how it is. But then they did not anticipate that the camel driver was coming along the way. Okay. But now, why did they ask these questions? Because they noticed these things. Okay, the grass was eaten only on one side of the footpath. Okay, that answered the first, this one. Where is that one? Loaded with honey on one side. Okay, this one, animal blind on his one eye. That is the answer to the first question. The second, ants loving fat buzzard on right side of the road. That is the answer to the second question. And then third one, with oil on the other, bees and other insects created on the left side. This is the observation which they made. Now, many of us do experiments. It's not just in chemistry or any branch of science. You do experiment, but what could happen is that which is not expected could happen. The only thing that we have to take care of is the way in which we observe that kind of a thing, the way in which we analyze that kind of a thing. That is what is important. That's what these princes were trying to do. Now, sorry about my English and all that. This is my English is not that great. For that reason, I just thought that I'll put this slide. This was a notice which was put in Chennai airport sometime back in 2019 or so. It was advertised in this one, NDTV. You see that eating carpet is strictly prohibited. This was shown there. Actually, what it meant was that eating on the carpet is strictly prohibited. Okay, this is just to show that sometimes we make mistakes in English. Just excuse me if I make any mistake in English. Okay. Now, what is this Prussian blue? This Prussian blue is a color that changed the artist's word. Okay, I have a sample of this one somewhere there. Okay, the color that changed the artist's word. The reason why this became so popular is because during the 17th century or before that one, people didn't have access to a good blue color. Okay, the thing that they were using was the ultramarines. They had to get it from Afghanistan and other places. Okay, but then it was really very, very expensive. So people were in the lookout for a good blue colored pigment, and this particular pigment satisfied all those needs. This is what I mentioned here. No, this Egyptian blue was there. Okay, this also silicate. Ultramarine also is a silicate that is generally found in Afghanistan, and then indigo is there. Indigo was not suitable for many purposes. Okay. Now, it is also said in some references that the Prussian army during the 18th century uniform, they had the this one Prussian blue on this one, but that is not true. It's probably indigo. But then in World War II, there was some use of this Prussian blue. German spies wrote secret words on their socks and clothes buttons when sprayed with ferrocyanide. Okay, ferrocyanide is there, and then when they spray with ferrocyanide, then letters called ferric sulfide reveal themselves in blue lines. That is Prussian blue or turnless blue. Actually, both of them have the same structure. Okay. Now, what is the origin of this particular pigment, which really fascinated so many artists around the world, and then for a large number of artwork also, this was really very, very important. There were two people mainly involved in this particular type of work. These are this Dippel and this one. Okay, and there is also another person, G. Stahl, who is famous for the phlogiston theory, and then Dippel version. There were different versions, but then the two people who were mainly responsible for this pigment, which was very, very widely used in the 18th and 19th century, were Dippel and Disba. That is what the story about that is what I am going to tell you in the next several slides. Now, this Dippel, okay, was an alchemist and a radical theologian. Many times he was jailed, okay, and then he was working in Berlin laboratory during 18th century, 1704 to 1707. Now, one thing that you should note here is that your even elemental nitrogen and many other elements were discovered much later. So these people didn't know what actually they were, they were doing as far as the chemistry part is concerned. So they were trying to mix certain things, but then in serendipitously they got this particular pigment. Okay, and then what he wanted to do was to find a universal medicine from the so-called animal oil. Okay, which was produced through distillation of animal or ox blood. Okay, don't feel bad, don't feel too bad about it, but that is the case. Okay, blood to which potassium carbonate had been added, and that used to, obviously you know that will give disgusting smell. And what are the types of compounds that it generated? Pyrroles, pyridines, nitriles. Nitriles are very important here. Okay, now, on one particular occasion, what happened was that there was a wrong label by his assistant, lab assistant. Many times you might have noticed, or if you are a teacher, you may notice that your lab assistant will mix or will give one compound for the other, and all those things will happen. Okay, such a thing had happened here. What happened was that there was a wrong label. There was a wrong label by his assistant. He wrote it as salt tartary, that is potash. Okay, potash is fine, but then he just wrote it. But the thing that he did not mention is it is from the waste material which was collected before. And then there is another person, the second person I am talking about, that he was a color maker, and he was in Swiss National working in the same laboratory around 1706 AD. Okay, this is between 1706 and 04 and 07. Now, what this person wanted to do was to get a red pigment. Okay, this Florentel lake, that's what is called, red dye carbonic acid. That's the compound, name of the compound, from crushed insects. Okay, in heated aqueous alum. Alum is potassium aluminum sulfate. Okay, to the filtrate, potash solution had to be added. So he needed the potash solution. Okay. To get the red carbonic acid. This was a paint in which this particular person was interested, and he used to get this one on precipitated alumina. Then what happened? Now this is the full schematic of that particular process. Okay, on those dyes, blood, all those things, destructive distillation, and then crude animal oil, multiple distillation, again distilled animal oil, multiple distillation over potash. Okay, that's purified animal oil, multiple distillation. This is the Dippel's oil, that first person's Dippel's oil. But then this he used to sell and saying that it's some kind of amrita or something like that. Okay, it's elixir weighted, that's what he used to call, and then he made a lot of money by selling this one. Many distillations of the crude animal oil, partially over potassium carbonate, and burnt light. Okay, it was a really commercial success. But then what happened there? The other person, about the other person I'm telling you now, the other person was using this synthesis to get this carbonic acid, which is red pigment. Okay, and that process is like this. Okay, that I'm not going to tell you anything about this one. He was using alum, insects he was using, but you notice that he was using potash at some stage. Now, he used to get carmine red Florentine light, red one, and purple colored material also used to get. But in both the cases, he was using potash. Now, what happened at a certain stage was that this Dippel was doing all these things. That's what I told you a couple of slides back. And then this Roser, which was the lab assistant, what he did was, there was the residue over there. That residue he labeled it as Roser's potash. Okay, that was the thing that he did. But then this particular person, what happened on a particular day, he didn't have enough of this potash. So what he did, he took this potash, okay, Roser's potash, okay, and then he added that one to whatever he was trying to do. Then the next morning, what they observed was this Prussian blue, which is a really dark, this one, blue pigment, okay, which became really very famous towards 18th century, as I told you before. So by the next day, he got this one, because there was so much of scarcity for low cost blue pigments during that time. Okay, they were able to sell this one. Okay, now this is the story about how the serendipitous discovery of Prussian blue was made. But the problem there is that this Roser, okay, he was his potash got exhausted on a particular day, and then what happened for this bar? He ran out of the potash that was collected by the lab assistant. Then he borrowed some more from Dippel. Okay, he also had potash. But then the problem there is that this particular thing, first time it appeared and all that, that's fine. But then after some time he ran out of potash, but ordinary potash won't work. Okay, initially what he did was to use the potash which was the residue from Dippel. Okay, residue from the animal oil and all that, which contains iron. Remember, that contains iron. Okay, but next time, once that got exhausted, he wanted to have this potash, but then what potash that he used that got exhausted, and now he has to use the normal potash. When he used the normal potash, it so happened that the ordinary potash won't work for that one. The reason being that, as I mentioned before in one of the earlier slides, the cyanide was liberated earlier. Okay, that cyanide from the animal blood or crucible, that with potash. Potash contains potassium, and that will form potassium ferrocyanide, and this will get oxidized by air, and then the color. It's a mist. About the chemistry, I will tell you a little bit later. Okay, but then he contacted many people for this one, but then the normal potash will not work. Then he requested Dippel, sir. Dippel's lab is where he was working. Okay, then what happened was that the calcination of potash with the dried blood, that's what they try to use now. Now what happened? Alkaline solution with cyanide and hexacyanoferrate to the acidic solution of carbonic acid, okay, and iron 2. Actually, carbonic acid is not required. They did not know the actual chemistry over there. Iron 2. Then they had the colorless ferrocyanide. This is the ferrocyanide. Okay, that got precipitated, but then it slowly oxidized by air to the deep blue, iron 3 hexacyanoferrate 2. Okay, this particular compound has both iron 2 and iron 3, 2 oxidation state, and that is the Prussian blue. So they got hold of a procedure by which they could get this pigment. Okay, and now, because as I told you, because of the scarcity of blue pigment, they made huge amounts of money. Some of the arts I am showing you later on, artists struggle to find an inexpensive, stable, brilliant blue pigment. That's indigo. There was a problem with that one. Okay, I will not tell you about that. Small azurite, all those, the ultramarine, and this is one of the things which they have to import. And this lapis lazuli, many of them are silicates. But then afterwards, there was a problem. This Dippel, as I told you before, he was a geologist. Then he used to give sermons and all that, and that was not liked by the king. and therefore, what happened? He was arrested for publishing the books against the Swedish king. Okay. Hence, to another, he has to be he was deported to he had to go to Netherlands and produce some Prussian blue, but then not so much. He was imprisoned. So, the whole thing stopped over there. But then this other person, the person who was making the pigment, that Disba, he met his teacher Frisch, okay, and they came to an agreement, and these two people what can I say knew now how to make a large amount of Prussian blue, and they called it as Berliner blue, okay, that is the same as Prussian blue, and they made huge amount of money by that. But then that secret was not out at that time. Now, there was another person who knew about something about this one, okay, and this Frisch or somebody, okay, they anonymously sent this details of this secret, how to make this and all that, okay, to this one, to this Neumann. He was a friend of Frisch, okay. This Frisch transferred his knowledge to Neumann, okay, and then this Neumann sent an anonymous letter to Woodward. Okay. This Woodward, what he did was, okay, he knew that something is working. He asked one of his colleagues to prepare that one. I think it is Jeffrey. Okay, prepare that one, and they knew that that kind of a thing does work, and that pigment can be prepared very readily in large quantities. But then he published that one in the philosophical transactions of the Royal Society. But then this person wrote the letter, but it was an anonymous letter, okay, and therefore the publication was there without the name of the author, okay, without the name of the author. But then that was in 1724. Now everybody knew how to prepare that one, how to manufacture this one. But you notice that here whatever you have to do is to kill the animals or take the blood and all that. This is so tedious, okay, but still the people who made the pigment got lot of money out of this one. Now the recipe is revealed, that is what I am trying to tell you. Okay. Now you see that this is dried cow's blood or ox blood, okay. Then you have to add potash, bring to ready, okay. This is the procedure. Procedure is fine, only thing is that they did not know the chemistry. And then blood this one leachate, okay, that mixed with alum or iron vitriol. Alum is not required there, that they did not know. Iron vitriol solution to the mix, they get a green precipitate, okay, and then filtration, pour hydrochloric acid over that one, and then over a period of 24 hours or something like that, they get the, where did it go? I am moving forward, just hold on for a second. I am moving forward, both sides are moving forward, which side is back? Okay, just hold on for a second. That recipe was revealed, okay. Now they got the Prussian blue, that is fine now. Now, which are the places wherein you can get iron, because now we know that we need iron for this one. How you can get the iron over there, and that also they did not know that iron was required at that time, but now they found out that that is required. And then gas purifying mass of the coal gas and coke production, in that one iron compounds were used to eliminate hydrogen cyanide from the coal gas by formation of hexacyanoferrate. Okay, that they tried to get that one like that. And then there was another process from methane and ammonia. This cyanide reacted with iron 2 to give hexacyanoferrate. The gist of whatever I am showing you here is to show that they have other sources of iron by which they can get the Prussian blue. Now this is Prussian blue, that is the color of that one, that first slide is there. Now this is a very famous painting, okay, art Vincent van Loo, the Starry Night, very famous one, and this is the other one, a Japanese person, okay, Hokusai, okay, Great Wave of Kanagawa. If you look at if you just Google it, all these things will come very readily. These two are really very famous paintings, okay. And then this one, Elizabeth Lewis, this is the this lady Teresa Countess Kinsey, this is another one, the black, this blue thing is from the Persian blue. And then unused blue 2 pence Mauritius stamp 1847, now it is worth millions. Okay. I hope that I had it, but then I do not have it though. Okay. But then crown jewel of philately, it is worth billions. Okay. Now, this is about the Prussian blue. You can also have green color and other colors if you have other elements or other metals over there. Green color, if you want to have the green color, mix Prussian blue and chrome yellow to depict a green garden sea. Now, overall there are different stories as far as the discovery of this Prussian blue is concerned, but there is we have to come to a sort of conclusion. Overall they say that it is around 1706 it was discovered. Okay. But then now, as I told you before, not much chemistry was known at that time. Okay. In 1771 Scheele found out about elemental nitrogen. This is 1771. You notice that this is in 1706, and then 1806 John Dalton hypothesis of the atom. Okay. 1807 Humphrey Davy potassium from molten potash. So, they did not know the actual elements which were involved involved in that particular process. And then 1782 Scheele and Gay-Lussac, they got the acid prussic, okay, that is the blue acid or cyanogen. This they obtained by the acidification of Prussian blue. Whatever you are talking about, whatever this hydrogen cyanide that you know of, that is that started from Prussian blue. Okay. And earlier it was called as prussic acid. Okay. Now, calcination of dried beef blood, okay, that contains protein, about 14% nitrogen, together with potash gives cyanic acid, formed when enough energy is added to substance, because they were heating it to really high temperatures, okay, containing carbon, hydrogen and nitrogen. Organic chemists should be happy about it. Happy or unhappy, I do not know about it, because casein is something which nobody likes though. Okay. Now, okay, the reaction is like this. HCN is produced, that HCN reacts with K2CO3 to give rise to potassium cyanide. Okay. Potassium cyanide reacts with plus potash, that is the Russell's Dippel's this potash. Okay. That was the that is the chemistry of that potash that they were using initially. And then overall reaction is shown here. Potassium cyanide is there. Now, with iron 2 plus you get potassium ferrocyanide. Okay, yellow prussiate on the potassium ferrocyanide, and then with again some more Fe2 plus you get this one. This is not that dark colored. Okay, this is not the colored one. Or otherwise you can add Fe3 to the if you oxidize that one to Fe3 plus, and then you combine that with the ferrocyanide. This is Fe3 plus and this is Fe2 plus. If you add that one, you get the Prussian blue. Here you have got both of them in Fe2, then one partial oxidation will give rise to Prussian blue. Okay. Fe2 to Fe3 partly, or Fe3 to Fe2 partly. Okay. One is called Prussian blue, other is called turbulence blue. As far as structure is concerned, both are essentially the same. Okay. Now, that is Prussian blue, and this is the insoluble version of that one. This is the soluble version of that one. Okay. Both of them contain Fe3 as well as Fe2. Okay. Now, you can also hydrolyze those things. Okay. If you hydrolyze, you get HCN over there, or if you in the basic condition you can get this ferrocyanide comes out and ferric hydroxide will precipitate out from that. Okay. Now, these are the things that you should know, and in this particular process you also know that should know that HCN is being involved. Now, this HCN is really very dangerous. Okay. I do not know how many of you know about this particular Holocaust. Okay. Here HCN was used to kill about 1.1 million people in Poland. Okay. That is, if you look at the Google, you can see that one. Okay. That HCN, the origin of that HCN is the Prussian blue actually. Now, as I told you before, these two have the same structure. The way in which you get the two pigments is Fe2 plus you add that to ferricyanide, or that is Turnbull's one, and Fe3 plus to ferrocyanide, that is Prussian blue. Okay. This that is the point that you have to, that is the normal thing, okay. That is how we know about it now, it is so easy. Now, after knowing all these things, it looks as though the chemistry was so simple, okay, but at that time they did not know about this one. Now, this is the other way by which you can write the structure. This is ferrocyanide, you see that is connected to carbon, okay, that Fe3 plus is outside. Now, overall structure looks like this, its soluble Prussian blue is blue, and then mu max is given there. Fe3 plus is high spin, this is for those people who teach the inorganic chemistry, high spin D5, Fe2 plus is low spin D6. Now, what happens in the structure is that Fe2 plus is connected to, this is Fe2 plus is connected to carbon end, and in the overall structure Fe3 plus is connected to the nitrogen. That satisfies our hard and soft acid and bases concept, okay, that most of you know about. The blue color, why does the blue color arise? Blue color arises because of the charge transfer between Fe2 and Fe3. Now, this is the structure, okay. I told you about that one. Fe2 is the red, okay. The red one, okay, is connected to the carbon, and carbon is this one green, okay. Fe3 is blue, okay. That is connected to the nitrogen end of that one. Nitrogen is red, okay. So, that is the overall structure that we came to know much later than what when it was produced. Now, at least the older generation know something about the so called blueprint, okay. Blueprint is a part of the story as far as the Prussian blue or Turnbull's blue is concerned, okay. It is something like what you have in Xerox now, okay. Earlier days, to make a photocopy of things, what they used to use was something, this one, the first thing that they used was to get the blueprint or cyanotype, 1840 to 1940. That is also an accidental discovery or serendipitous discovery, okay. This person in 1842, first ever industrial photocopying process, multiple versions of the original, I mean, okay. Using Prussian blue or the Turnbull's blue, okay, useful for architects, okay. I do not know how many of you are that old, I think only older generation will know about it, okay. Anybody older than me, more than 70 years, nobody, okay. Earlier generation will not know about it, okay. Only I think he will know about it. All right. The name is derived from this one, blueprint is that. Now, what they used to do was, okay, the paper is coated with potassium ferricyanide, okay, and this one ammonium iron 3, okay, iron 3 citrate, this is citrate, both are soluble. The second one, what happens, undergoes photo reduction of Fe3 under UV light and reacts, and then what you have to do is hold a black ink sketch drawn on tracing paper or photosensitive paper and expose them both to light source. This is the first Xeroxing, so to say, okay. Parts that are not protected by dark blue lines, okay, will change their chemical formulas in the light, or not protected, what will happen, there you get the Prussian blue or the Turnbull's blue, would change their chemical formula and the light, and from ammonium ferric citrate to ammonium ferrous citrate, then Fe2, this is Fe2, it has become Fe2, okay, then Fe2 you have got the here, and then that will react with ferricyanide, okay, to get white sketch, okay, like this, white sketch on blue paper. It is light stable, now familiar term blueprint, okay. Many times you use this term blueprint, but then that arose because of that. Prussian blue was also, this is just an addition, that is all. It was also called as the molecule, it was the molecule of the year in 2017. Now, how many of you recognize this one? Anybody recognizes this one? Blueprint, okay. Earlier days people used to have this kind of a thing. The reaction is this one, reaction I have given you there, per Prussian or Turnbull's blue, okay. That is the kind of sketch which people used to make for making the drawings of the buildings they are going to construct and all that. Now, this also, there is also a possibility that you can have the smart windows using this Prussian blue, but then the ones which are currently being used are not from the Prussian blue, okay. They are from a different source, okay. The more common is the tungsten, this one tungsten dioxide, halogen, etc. But then this also can be used as switchable windows, okay. And then the original crayon color and other things, the people were using this Prussian blue, and this also still it is used in pathology tests for bone marrow and all those things, thermal cancer treatments and all that. And the other major use here is antidote. It is an antidote for heavy metal poisoning. You may remember something about Saddam Hussein, and then radioactive cesium. You may remember something about Brazil accident and Chernobyl accident at all. And in such cases, okay, this Prussian blue can be utilized. Take out the, what can I say, radioactive material, okay. And histopathy stain for detecting iron. Prussian blue batteries, lot of work is going on there. And then Prussian blue stain, 1867, it was there for the detection of iron in tissue. Some of these things are still being used, okay. And then towards single molecule magnets, that is another paper, and there is an extension also of these things. Now, it is not necessary that you need to have iron 2 and iron 3. You can have other transition metals. When you have other transition metals, you can have lot of other different colors and all that. That also has been investigated really thoroughly, okay. And then there are this is from my colleague Radhakrishnan, that is why I just put that one there. Lot of other people also have done that, okay. And but then there is also a dark side to the story of this one, HCN and all that. HCN is just hydrogen cyanide, which originally got from the Prussian blue and all that. There is bitter almond smell is there, you know. The speech beads were used in judicial executions, okay, as far as back as ancient Egyptians, okay. Ancient Romans used to use the cherry laurel leaves as an execution method, cherry death. And what was the possible ingredient? That was cyanide over there, okay. In all those things, that where did the cyanide come? Originally from the Prussian blue and all that, okay. And this Scheele, I think most of you have heard of this person's name. He mixed Prussian blue with dilute sulfuric acid, okay. He was able to produce a new gas that was colorless, water soluble and acidic. We now know that this gas was is hydrogen cyanide, as I told you before, okay. It is called this one dark blue, or the Blausara, okay, or prussic acid. Only thing is that don't try to do that one, because this acid is really very poisonous, okay. Although Prussian blue is not poison at all, but this is poisonous, okay. And then this one in England also something had happened there. On 1845, a neighbor found Sarah means this lady, okay. She essentially died, but then the attending doctor had followed the man. There was a man along with her, okay. And then this man, what he did was to give her this one cyanide, and then he he just left. But then earlier days, you know, you didn't have this WhatsApp and other things, okay. They sent this electric paragraph, and finally they could catch hold of that person. And the story is that this lady was killed by means of that kind of a cyanide which originated from that one. He had purchased a bottle of prussic acid shortly before he visited, and so they were able to catch him, okay. And I told you about the gas chamber in the this one Second World War and all that. And then Jean Lewis, it is said that there was some problem as far as his this one is concerned. This hydrogen cyanide might have been responsible for the problem associated with his death, okay. Now the references are here. I don't know how much time do you have? 5 minutes, 10 minutes? 2-3 minutes. I see, okay, that is fine. So then I will not go too much to the ferrocene story, okay. And you see that these are the ones. These are two things I have already shown you, and this another one, Prussian blue type nanoparticles and nanocomposites. And then the third one is, this is that lady Anna Atkins, 1843, okay. This is how she got a lot of them really printed. It looks as though it is really real, okay. And history of ferrocene and all that, I don't know whether I should go to this one or not. If I don't have time, I think that I will stop. Pardon? 10 minutes I have? Okay. All right. Okay. If I have 10 minutes, I will just finish it in 10 minutes. No problem. Okay. Now, this is purely chemistry. Now, the reason why I chose this one is because after this ferrocene was discovered, the so-called organometallic chemistry flourished like hell. Okay. But then before ferrocene was discovered, then also large number of organometallic compounds were there, but then that did not what can I say, that kind of a literature did not provoke people to go for what can I say, too much of work, this one scientific work or chemistry as far as the organometallic chemistry is concerned. Now, the reason why I chose this one is because people were interested in this particular compound fulvalene. Okay. It was predicted to be aromatic, okay, and therefore, what this Pauson and Keeley did, they were doubtful about that one. They were trying to prepare this one and then oxidize this one, because ferric chloride can act as an oxidizing agent. This is what they do. That was their logic. They wanted to prepare that one. Okay. Then what happened was that they got a product which was orange solid, and all the properties are given here. Okay. The reasoning I told you just now, ferric chloride will be okay, initial reduction of FeCl3 and then followed by the reaction and all that. This was accidental. They did not anticipate this particular thing, this particular type of reaction over there. But then the credit goes to this Pauson, because his paper got published. But then before his paper got published, okay, these people had submitted the paper. Okay, they had this compound before, they submitted the paper, but then the publication became late over there. But then even before Pauson is the one who is being credited for the isolation of ferrocene. But now there were workers in Union Carbide, okay, they had some yellow sludge. They were just passing iron through this cyclopentadiene. They had yellow sludge, and then that was blocking the what can I say, flow of the gases, and they didn't know what to do with that. Therefore, they just discarded that one, but they discarded, but collected that one in a container or something. They had that one. Okay, but then the original, the first literature appeared here. Although the submission by these people were was done much before that one. Now, there is a lot of story about the ferrocene also. Pauson I told you, he was a refugee from UK, and then the thing is that in ferrocene, if you look at the structure, it is a sandwich type of structure. Okay. About the proposal of that particular structure, there was a fight as far as with the Nobel Committee which involved Woodward also. Okay, Woodward, and then the structure was proposed by Woodward, and then Wilkinson should be there. Wilkinson is there, and then Fisher, Pauson attended some meeting, but then in that meeting, okay, somebody told him about this one. There is a possibility like that, but this can be verified by using magnetic susceptibility and all that. This is something which happens to most of us, because sometimes we are ignorant about certain parts of chemistry or certain parts of science, and then what will we ignore that one, and we will not know that we have missed a really great chance like that. And Pauson missed that one. Somebody told him, but then he missed that one. But the type of structure, that is the ferrocene structure, was proposed by Woodward, and Fisher also found out that one. Okay, they said Doppelkugel and Woodward and Rosenblum. Okay, they proposed structure based on the magnetic susceptibility. At that time there was no NMR. Okay, magnetic susceptibility IR 1 CH stretch and dipole moment 0. Okay, that is how this one. But then here is the story about this one. When you want me to stop, you stop me, no problem at all. Okay, but then the story is like this. When Nobel Prize was announced, and Nobel Prize was given to not given to Woodward, it was given to Wilkinson as well as Fisher for developing the organometallic chemistry. But then Woodward wrote to the Nobel Committee, okay, saying that I was the first to propose the sandwich structure for these things and all that. Okay, he said that one, he essentially tried to fight with them. Okay, saying that I should also have been given the Nobel Prize for this one. He already had got the Nobel Prize, but then this also was, he proposed, there is no doubt about it, that they did not bother about that one. They said that I do not know whether they answered that one or not. Okay, but then he did propose that one along with Wilkinson. Okay, but there was a problem of that type. All right. Is it time? Okay, 5 minutes. Okay, now the original structure that was proposed is like this, but then the actual structure which was finally proposed by Wilkinson, Woodward and Fisher was this one. Now, how did this one Pausen miss that one? What he did was he had the crystals. There was another scientist to whom he gave the samples, but then that scientist was going around. I think it's Jacobson. He was just going around, so the structure was not done by him, and his sample was not investigated further. But then these two people, Wilkinson as well as Woodward, got to know of it. Woodward proposed the structure based on the IR, single stretch, IR, but then Fisher proposed it on the basis of other measurements and all that. Okay. Now, why did they get the Nobel Prize and not the Woodward? The possibility is that, or the explanation is that Fisher and Wilkinson developed this organometallic chemistry out of this ferrocene. Instead of ferrocene, they Fisher prepared the dibenzene chromine and many other things which are analogous to that one, and hence a huge amount of work was done in organometallic chemistry, not synthetically. It's also application oriented thing, and because of that one, those two people got the Nobel Prize. And then between Fisher and Wilkinson, there was always a fighting. They divided the periodic table, but then the students won't listen. They will trespassing with each other. All those things were happened. Fisher was upset when Wilkinson and his German speaking co-worker F.A. Carton's book, I think all of you know about it. They wrote a paper in German also. And then Wilkinson to Rosenblum was a student of Woodward. Okay, from Woodward, he asked. Okay, Wilkinson was very unhappy with him, saying that why do you want ruthenium chloride? Because they wanted to prepare the ruthenium analog of that one. And then, as I told you before, Woodward also wrote to the Nobel Committee and all that. Okay, but then nothing happened. Only these two people got the Nobel Prize, Wilkinson and Fisher, and not Woodward for that one. And Woodward, what he wrote here, both of these concepts, this unusual sandwich structure and prediction that such structures would display unusual aromatic characteristics, both these concepts were simply, completely and entirely mine, and mine alone. This is what Woodward wrote, but they didn't bother. They didn't give him the Nobel Prize for that one. Okay. And then these things I will not go into the details of that one, because I just wanted to tell you something about ferrocene and all that. I will perhaps go to this one. Okay. No, these things I will leave out. Okay, because I think that I had to leave some time for the questions. Okay, and this is perhaps the last slide. And I did not tell you anything about my research work and all that. This is part of my research work, just one slide for the sake of showing it, that's all. Okay. Overall, what I wanted to tell you is that there are a large number of discoveries, not in chemistry, okay, in all sorts of science and all that. The only thing that we have to know about is when we observe something unusual, this is for the younger generation, when we observe something unusual, what you have to do, you have to try to analyze that one. You should know the significance of this particular observation. Now, the thing is that this as far as the organometallics is concerned, Pausen, okay, he sent that paper to Nature, whereas the other three papers sent it to Journal of this one Chemical Society, okay, because they knew that it was different, but then they did not understand the significance of that particular compound to that extent. The reason being that they were unhappy that the iron compound they got was not able to catalyze the reactions they wanted to do. So, they just lost interest, they just wanted to publish it. But this Pausen knew that there was something really very, very unusual about this particular compound. He sent it to Nature and he got a good publication. The unfortunate thing for him is that he didn't extend that one because he was not having a permanent position, and they didn't recognize his work as far as the Nobel Prize is concerned. But then his publication got in Nature, the other people, this one they publish in the Journal of Chemical Society. This kind of a thing happens to many of us, I think, those people who are working well versed with publications and all that. Okay, thank you all.
Thank you, Professor Kumaraswamy, for an excellent talk on the discovery of protein and its protein Persian blue and then its users. So, open to a couple of questions. Questions from students?
It's actually a very good talk, and this Prussian blue is replaced by the bimetallic like bismuth or some other.
Yeah.
So what the main reason for the color in this case actually?
In the other cases and all that. In the bismuth case, it's a little bit difficult to say because bismuth has two oxidation states, bismuth 3 and bismuth 5. But the color variation, I think that there may be some other reason also. Although in those cases, bismuth 3, I don't think that it will go to bismuth 5. For bismuth, the normal oxidation state that we know, bismuth 5 is known, lot of work is there, but then I don't think that gets oxidized or something like that. So the bismuth 3 could act like this iron 3, that is a possibility. Okay. But then bismuth 3 has other features and all that. Now you many of you may know about the perovskite and other things. That is something that I am not aware of that. Bismuth based perovskites, they have some other interesting features. There is a lone pair over there on bismuth.
Any questions from students? If there are no questions, let us join me thanking Professor Kumar. And let's all take a coffee tea break for 15 minutes and then assemble back.
Finished in perfect time.
Perfect. Thank you.
Professor Ganesh, please remind me 10 minutes before the end.
Yes.
Just tell me,
Sure.
so that I know.
Yeah.
Okay. Can you hear me? Can you hear me at the back? Okay. I feel really privileged to come and give a talk here. A lot of effort has gone by many people who has organized this, primarily to see that people, school teachers, school students, college teachers, all see a benefit of panorama of various subjects. So one such thing that I am going to do is discuss dimensional analysis in biology, chemistry, physics and medicine and things like that. But before I start, I am nostalgic about this place. I have been here for a very long time. The first interaction I had was I was in the main guest house here for some meeting or attending some lectures. So for breakfast, I was young then. At breakfast, I asked for some coffee or fruit juice, I forget. And then I was impatient. So I asked the person who was serving me, please give it to me in a maybe impatient voice. So he turned around and said, sir, I have served people like Professor C.V. Raman here. So that put me in place. I realized how big a place this is, how historically it's a big place. Secondly, I know several people here. One is starting from H.R. Krishnamurthy, with whom I have worked, I have done some work. And then after that, Arnab Rai Chaudhary, then Sri Ram, Deeptiman Sen. I can go on naming people here. They are all my friends. Here Usha Devi I had the privilege of knowing now, and I have learned so much of quantum physics from her. So now let me start with the beginning. Here, just a minute please. This dimensional analysis can be applied to almost any place. The way we start understanding dimensional analysis is the following, that we have mass, which we all know, length and time. And these are the only ones. Can everything be derived from here? The answer is no, it need not be derived from here. Energy can be derived, momentum can be derived, and velocity can be derived and so on. That's one. Maybe there are other areas which we should look at. One of the areas which has been looked at for the last 175 years or so is information. If somebody told you information is form of energy, you would say this guy should be put in the madhouse. But that is no longer true. It started with Maxwell, with Maxwell's demon. Remember that when Carnot talked about Carnot's engine, at that time he did not know heat is a form of energy. Heat being a form of energy now we are told in schools, so we will accept it. If somebody asks us to prove why is heat is form of energy, it will take a lot of effort. So that's the first thing, and it did take a lot of effort to find out why is information form of energy. Now after saying heat is a form of energy, you will say how much of heat will I equate to calories or joules. There is a famous experiment in which you keep water and go on stirring it, its temperature will increase. So you know, using the specific heat, you know how much energy. So this is the famous 4.2 times some number of joules per Kelvin. That's the answer. Can we do that for information? The answer is yes. People have done that. So this is one. I will give you an example which to my knowledge has not been completely answered. I am not an expert, so I won't know. Say take emotion. Today you would think emotion is not a part of any scientific thinking. We don't know. I mean, if you look at the history, things change. So I am like to keep it as broad as possible. And today, for example, when you were giving a talk, you were mentioning about bacteria. I know that in diabetes you have sugar level numbers. So are there numbers for bacteria? And questions like that can be asked everywhere. Like in chemistry also you can ask the same kind of question. Maybe they are being asked. So I don't know. So what I have done is taken some simple examples and work my way through. So, that is the object of this talk. Is that okay? So, let me start. So, I have written here various possibilities: biology, chemistry, physics, medicine. Again, I have written medicine. One was the heart, the other one is some drugs intake, and of course, mathematics. All these are there. Let me proceed further. The first thing I will do is in mathematics, is a well known result, is the Pythagoras theorem. Let us look at this triangle ABC. Is there a way in which, oh sorry, how do I go? No. Oh, how do I go back? That one, first one.
This is back, this is front. This point.
Which one? This one.
No, this is
Yeah.
front.
No, but I want to have I use it as a pointer also. Thank you. Which one? This one? This one?
This one.
Okay. Okay, here is A, here is B, here is C. This is the triangle. Now, area of a triangle is length squared. So, I have just decided to choose CA. Oh my God, what is happening? Okay, I will just go ahead and show it like this. A area is C squared is length and some angle, this angle is phi. This angle is phi. Now, look at this triangle. I have two other triangles, ACD and CDB. The angle DCB is also phi. Why? Because that is phi and that is 90 minus phi, and triangle ACB is a right angle triangle. So, angle CDB is phi. Is that okay? Now, I use the same logic there. The area of A1, A2 has been displaced, I do not know why, d squared g phi, and A2 is c squared d phi. A2 is c squared d phi, AB is d squared d phi. So, now when I add the two, I get A squared equal to d squared plus c squared, which is of course the Pythagoras theorem. So, I have not used any other geometry, just scaling to get to the Pythagoras theorem. Is it all right? Let me go to the next one. Oh, I am going the wrong way. All right. This is height versus blood pressure in animals. Again, taller animals need higher blood pressure to pump blood to the head. This is all very trivial, but still it is important to realize how dimensional analysis is used. The heart must pump blood to head against gravity. Some quantities that may be important for the problem are the height of its head above the heart, h, the volume of the blood pumped per beat per unit of beat, stroke volume, Vs, the density of blood, rho, the acceleration due to gravity g, and as well as the measure of animal's blood pressure. These quantities come from your experience. If you are not experienced, you may miss some quantities or add some irrelevant quantities. So, that is part of doing dimensional analysis. So, now I do the h is dimensions of L, rho is m by L cubed, g is L by t squared, volume is L cubed. So, you can use dimensional analysis to find out the pressure is mass, force per unit length area, so that you can just calculate and you will find this out. Now, there are 5 variables you can see 1, 2, 3, 4 and 5 variables, and there are 3 dimensions m, L and t. So, you will get 2 dimensionless constants. Okay. So, these are the 2 dimensionless constants here. Now you use the knowledge of medical physics to get pi 1 is equal to s pi 2. and then you get the result, which is nothing very great, is pi 1 is proportional to P by L. P is proportional to H, which we teach in middle school. That's pressure is same thing as height. I just took this example as a matter of dimensional analysis. I will go to more difficult examples. So, P1 by Ph is equal to this, and then I go next. P1, P2 being the pressures and H1, H2. Since the animals like giraffe have long necks, about 7 times that of a human being, the blood pressure of a giraffe is about 7 times more. This calculation is simplistic and is done only to show the power of dimensional analysis. A more realistic calculation needs to consider the viscosity of the blood, radius of the blood vessels carry, and so on. This has been done, people do this and they get more complicated results, and they all agree with experiments. So, without knowing too much theory, they can get some results. Okay, that is how the whole thing goes. Okay, I got all this slides from Professor Saurendu Gupta of Tata Institute. So, though I did not formally acknowledge here, I am just saying that here. Okay, dimensional analysis and architecture. Now consider how high can we make pillars given a cross sectional area A. If the pillar is too short, too tall, it can fall. If it is too short, then the pillar can be made thinner and the cost can be reduced. So, what is the important quantity, physical quantity to be considered? Certainly the mass load, the pillar has to support MP, the area of the pillar AP, and the density of the pillar rho p. One needs a relationship of the form HP is the height of the pillar, some dimensional, some relation between MP, AP and rho p. Okay. These are the things which you are imagining will be there. Of course, there could be more complicated ones like earthquakes, what will happen and so on. We will not get into those discussions. Mass of the pillar is m, HP is L and AP is L squared. Now, we have 4 quantities. So, out of the 4 quantities there are 2 dimensions. So, we could have 2 possible dimensionless quantities. They are pi 1 h by root AP and pi 2 pi a to the power of 3 by 2. Now, there is this Aireteshwara temple near Kumbakonam. I have seen this temple and it is pi 1 is equal to 15. Ashoka's lion column from Sarnath built 300 BC as pi 1 is equal to 15. The most impressive solid rock pillar in the world. So, we could build things like that at that time. Okay, now let me go to the next one. This is something which I teach in almost all classrooms, and those of you who are teaching physics, chemistry, please look at this carefully to see what is it all about. We start by, these are for dimensional analysis in atomic, molecular and biological system. We start by noticing the dimensions of velocity of light, the electric charge and the Planck's constant. The velocity of light, of course, is velocity, it is L by T. The dimension of the electric charge can be got by noticing the relation Es is equal to E squared by 4 pi epsilon 0 r squared. E is the electric field. This is the standard expression here which we write down, and I am working in SK, what is the set of units which we use? SK some. No, the standard units you use, I will just say standard units, that skips me, and then energy is nothing but E squared by 4 pi epsilon naught r. E is the electric field due to 2 equal charge E, Us is the energy stored in the 2 charges, separated by distance r. Finally, epsilon 0 is the permittivity of the free space. Thus, the dimensions is E0 by 4 pi epsilon naught. So, I can immediately calculate the dimension of E squared by 4 pi epsilon naught, that is ml cube by T squared. And E we all know is given by h nu, which is the Einstein's relation to understand photoelectric effect. Incidentally, there is a very interesting story about Einstein and Millikan. When Einstein had made this suggestion of E equal to h nu photons or particles, at that time, please go back to this time when electromagnetic wave, a continuous thing, was explaining everything. There was nothing that was missing. So, when Millikan said, he said this must be wrong. So, he did the experiment on photoelectric effect and checked this, and then he writes saying, I tried my best to prove Einstein wrong, but I was proved wrong. And then of course, Millikan got a Nobel Prize for this. And so, that is this. Now, E is the energy of the photon and nu is the frequency of the beam. So, h is energy. If I had put a T squared here, it would have been energy, but I have to multiply by time. So, I got rid of that. Using this relation, it is easy to show that hyperfine structure constant alpha is dimensionless. Here is the famous suggested by Sommerfeld, E squared by 4 pi epsilon naught Hc is 1 by 137. Now, consider the radius of a hydrogen atom. Bohr's orbit, we teach them in schools, and class 11 or 12 we teach it, and it is given by h cross squared by me squared, and I have to put a 4 pi epsilon naught here to get the units, and n squared, n is the principal quantum number. So, n equal to 1 is the lowest orbit. How can I get this? I can get this by looking at this. Firstly, look at this quantity h by mc. If I calculate the dimension of this, the dimension of this is length. I will not do the calculation, you can do it. It is called the Compton wavelength. And here, but hydrogen atom, Bohr's hydrogen atom is non-relativistic. If it is non-relativistic, velocity of light should not enter at all. So, I have to get rid of C from this equation. So, how do I get rid of C? I have alpha here which has a C. So, I am divided by alpha, C will be in the numerator, so it will be cancelled, right, it will be cancelled. So, the correct answer is AB is equal to this expression which I get, and then you can do the dimensions of it. Now, let us talk about the velocity of the electron in the ground state. With the velocity, one velocity is c, but c should not be there. So, I divide c by r, I multiply c by c alpha. Oh, what did I do? Where am I? Here it is c alpha. So, immediately we get an idea that the velocity of electron inside is 1/100 of the velocity of light. So, it is a picture that you should keep in your mind. Then now I can calculate magnetic moment, various other quantities simply by using dimensional analysis. Is this clear? This is something which I use in a classroom, because when you teach physics or chemistry, getting the order of magnitude is very important. Whether you are talking about a needle or you are talking about an elephant, you will know what approximations to make. That is crucial. So, this is another thing which I do. Now, this is a very famous example of the fluid mechanist Taylor. There is an atom bomb which was exploded in somewhere in New Mexico, and it was a secret result done by the Americans before they hit the Japanese with 2 bombs, and they did not reveal how much was the energy released. Then they did not tell British also. So, the British defense people contacted Taylor and wanted to, before I have written down something there, I have skipped that, but anyway, that does not matter. Now, this is the general picture. I have here an atomic explosion taking place, a red spot, and then the moving air. There is a shock wave front which is moving, and atmosphere is outside. The energy released in the atomic explosion, the Taylor assumed it should be proportional to r, the radius of the explosion, density, and the time. the r is the radius of the shock wave, t is the time, and rho is the density of the surrounding air. So, now you calculate this, E explosion is equal to k r to the power of x, rho to the power of y, t to the 2. It is quite straightforward to do the algebra. I will skip the algebra here, and the answer is given here is k r to the 5 rho t to the minus 2. Taylor knew beforehand by looking at some other experimental data k must be of the order of 1. In 1947, a view of the explosion was released to the public. So, Taylor got hold of that article and noticed the following. He said that when t is of the order of 1 second, r is of the order of 100. So, if t is of the order of 1 second, r is of the order 100, rho, the density of the air he knew, he could immediately calculate the expansion, the explosion. When he declared it, of course, the American government said that this is not right, they did not want to tell the truth, but it was right to within about a factor of 10 or so. But that itself shows how powerful dimensional argument plus your knowledge of the physics or whatever be the situation involved. It is not for the layman, it is Taylor who knew a lot of experimental details where he could figure this out. Okay. The deflection of a beam under a load. The deflection delta of a beam depends on an applied load, beam geometry, material properties and boundary conditions. For simplicity, we assume the supported beam with a point load and uniformly distributed load, as these are the common cases when deflection formula from Euler-Bernoulli beam theory, which we do this experiment in undergraduates in physics, is given by this. The goal is to find a relation between these. Maximum deflection is given by that, applied load is F, Young's modulus E, moment of inertia I. You can write the dimensions of all that. I will skip again the dimensional analysis and you get this. This one was given to me by a lady, a civil engineer, Lisha Varaske, and I think that is all that I have to say. I thank Rajaram Nityananda for suggesting the talk about dimensional analysis. I have always valued his judgment, and several academics. I also thank Ritesh Bharat. I thank this for suggesting this, and of course I thanked Saurav Gupta. By the way, I talked to Satyamurthy, who is a chemist, most of you know him. He has also given me many chemical reactions based on dimensional analysis. So, I will stop here.
Thank you very much. I just wanted a comment on the dimension analysis in the case of velocity of an electron inside.
Okay.
So it is non-relativistic. But I got reminded of the other one, but what we do as Vyskov estimates, where one looks at proton also.
Yes.
Protons.
Yes.
Protons also have less than the velocity, again using dimensional analysis.
Correct.
So that is also one thing that I got reminded. But that gives you a question on why do we do relativistic quantum mechanics then?
Why?
Relativistic quantum mechanics and hydrogen atom. So
Yeah.
this may mislead.
No, if you do it relativistically, the formula is very complicated. So, I do not know whether it can be obtained by dimensional analysis, I have not seen one. Magnetic moment can be calculated. Thank you very much. Thank you.
So let me invite, my pleasure to invite the last speaker, Sir Dr. Maya Rao from Astrophysics, Department of Astrophysics, Raman Research.
Oh.
Um, all right, first of all, thank you. It's a delight to talk to a very varied audience. So I've planned this talk to address students, teachers who have interests across disciplines. So if it's too basic for you, I apologize, but hopefully towards the end you will see where I'm going with this. The focus of my talk will be to give you a flavor of the kinds of techniques we would use to study something like the first stars, and towards the end I will get to the part where I will discuss the work that we do at RRI about this. So let's start with having some common ground about what is a star. I think we all have an intuition of what a star is, and most of us in this audience know that there is something about nuclear fusion, hydrogen, helium, something, right? There is a governing body, a consortium if you call it that, called International Astronomical Union, IAU. They were the guys who kind of kicked Pluto out of the 9 planets, right? So they have a definition of what a star is. I'm just going to paraphrase to my best memory, okay? So it is a spheroidal plasma, a luminous spheroidal plasma that it holds itself together with gravity, sustained by nuclear fusion at its core, okay? That is a all-encompassing definition of what a star is. But we know if you look out into the night sky, hopefully outside Bangalore somewhere on a highway, you see a bunch of dots that are bright, and we know those are stars. We also know that the sun is a star, okay? And stars have different classifications. We won't go into that today, but for the most part, what we call in astrophysics parlance a main sequence star burns hydrogen through nuclear fusion to give you helium. So once it burns all of its hydrogen to helium, the helium burns to carbon, carbon to iron, and there are a bunch of processes. Some stars explode in supernovae, some become something called brown dwarfs. There are different kinds of stars, okay? But that's what a star is for all practical purposes today, okay? Now, here are some numbers that just to get you thinking, okay? We have different measurements, including something I'll discuss later, that have told us that the present age of the universe is about 13.8 billion years, okay? So we also think our sun is about 4.8 billion years old, okay? Which means that the sun was born at some point of time in the history of the universe, right? That's common sense logic, which means that the sun and the universe are not of the same age, right? And the sun and maybe stars older than the sun were born even earlier. Okay, so now I hope we agree that stars are born, they have not always existed. So first question, how do you think we know the age of the sun to be about 4.8 billion years old? I gave you all the answers to get the question, right, to this question. We know how much hydrogen has been burnt into helium if you take a spectrum of the sun. That's just one technique. We can also estimate typically the age of the solar system, okay, by looking at, for example, moon rocks and seeing some radioisotope measurements, like how we do carbon dating on Earth, we can do other radioisotope dating of different parts of the solar system. Earth is a little problematic unless you go deep into the core because there's a lot of other contamination, right? So we have found a way to get the age of the sun, okay. Now, with a lot of the measurements through several years and a lot of theory, we have arrived as a community in astrophysics. For the most part we agree, there are some exceptions, that we live in a universe that we call as part of a standard cosmological model, okay. In technical parlance we call it the lambda or dark energy cold dark matter, lambda CDM model, okay. In this model, at some point we don't know exactly what happened, and that's a big question mark. The universe starts, okay, there is a T0 in some singularity. In popular language that's called the Big Bang, okay. We don't really think there was a bang, it was not an explosion defined in space and time at one point that expands. Everything starts at that point, okay. So let's not try to think about what was behind that point. At some T0 your clock starts ticking. Within the first few minutes, all the matter that you and I are made up of today, like all the atoms you think of, this table, laptop, all the particles, we call them as baryonic matter, okay. The type of matter we don't understand but we think is out there is something called as dark matter, okay. There's something else called dark energy, and there is energy like you and I understand it, right, heat, light, right. So we'll call that energy. So the universe is filled with dark matter, keep that aside, dark energy, keep that aside, but we know there were baryonic particles, okay, and light the way you and I understand it, okay. This baryonic matter is very, very hot, it is a plasma, okay, because the universe has started out in a very compact place, okay. So the baryonic matter exists as protons, electrons, some helium nuclei, okay, little bit of lithium somewhere, and that's pretty much it. So the early universe had only compositions of hydrogen and helium, but it was not in the form of an atom, it was in the form of ionized plasma, okay. And the light or the energy kept bouncing off of these ions, okay. So it was what we call an optically thick medium, okay, it is opaque. The light cannot escape the interactions from these electrons and protons. Gradually the universe expands, which means it also cools, and the first atoms can start forming. Okay, this period when the first atoms form is called as the epoch of recombination, and we think that happened at about 380,000 years, about 400,000 years after that T0. And this light that was bouncing off of these ions is finally free to escape from this interaction and comes towards us. So today we are able to receive this light from the very early universe, and we call that light the cosmic microwave background radiation, that really colorful green-blue layer, that is the CMB, cosmic microwave background, okay. So if your eyes were sensitive to microwave radiation, if you don't have an intuition, that's about 100 gigahertz or so, okay, in frequency, the sky would be aglow with a light, okay, and it's a very, very weak light. The temperature of that light is about 3 Kelvin. If you think of it as a black body, it's about 3 Kelvin. So if you go to outer space and you want to know what the temperature of outer space is, it is about 3 Kelvin, because the entire universe is filled with that microwave background. Okay, let's move on. So that happens to the light. What happens to the matter? We know that at some point the first stars have to form, right? Because today when you look out into the universe, the first stars are there. There are some stars, but at that point there are no stars and galaxies. At some point, and this is where the problem comes, we don't know exactly when and we don't know how the first stars and galaxies in the universe formed, okay? And that's what this talk is about. How do we go about trying to study those first stars? Okay, first of all, why is it important or interesting? Well, to me as an astrophysicist or cosmologist, it's a big piece of the missing puzzle. Like, you don't know what's going on. If you can't explain that part, your model of the universe is incomplete. Okay, for me it's interesting for that. But remember, it is these first stars that then explode, create the next generation of stars, to create the carbon that you and I are made of, to create the silicon this laptop is made of, okay. There was a starting point that started all of this, and these stars are key to that, okay. That's a more philosophical reason why it's important to understand. Okay, I won't spend too much time on this, but for those of you who have not seen this, please go take a look. I don't think we can appreciate how long 13.8 billion years is, okay. This is called the cosmic calendar. The Big Bang, T0, is on midnight, 0.00 hours of January 1st, okay. We are right now December 31st, midnight, okay, almost coming to the next year. So the entire 13.8 billion years is put on this calendar of 1 year. To give you a context, the first human beings, as we think, right, the early first Africans who migrated out, right, they came on the picture in this calendar on the 31st of December at 11:52 p.m. So humankind has been in existence for 8 minutes in the entire year. That is how old the universe is. I hope you can appreciate that context now, okay. We are trying to study February, okay, when the first stars formed. But did the first stars form on February 1st, 28th? What did they look like? How big were they? We have no idea. That's the problem. Okay, so I've just summarized the same thing. We don't know anything about the first stars, and broadly this period in the history of the universe is called the cosmic dawn, okay, because it sounds nice, it's appropriate. We think approximately it happened when the universe was about 100 million years old, okay. So like I said, February. Now, one obvious answer to this question is you build ever more sensitive telescopes. So you and I can see some things with the eye, we can see some dots, some stars. Maybe we can, if we observe close enough, we can see some Andromeda. You go to the, you see some point galaxies, right? But you can build a really huge telescope, and I think this is something everyone is aware of, the James Webb Space Telescope, right? This is the current most sensitive telescope, and it's in space. And this is what's called a deep field image, okay? So this, I don't know the exact number, but it has about some 30,000-40,000 galaxies. These are not stars. Each dot you see on there is not a star, it's a galaxy, okay? And this has been imaged by James Webb. So what is the furthest James Webb has seen? Does anyone know a number? You can throw a random number out there. You can tell me either in something what we call redshift or in terms of age of the universe. Any guesses? Close, 14 point something, okay? Yeah, but good try. That's the redshift, about 14.1, yeah. So here I put a bunch of questions. So I don't expect you to answer them, but this is what you have to think of, right? Because this is the most intuitive way to do it. You build a bigger and bigger telescope. So how big is the mirror of James Webb? Because that is the best telescope we have now in space, okay? We need to, what is the farthest James Webb has seen? We said it in terms of a redshift, we will convert that to years. So how far do we need to see to go to study the first stars? Is it right? Can we see it with James Webb? And if you can't see it with James Webb, how big a mirror would you need, okay? What is the biggest ground-based mirror that you have or it's upcoming? So here are your answers, okay? So James Webb has a 6.5 meter primary, okay? It's an optical telescope, so we all think, we have an intuition of optical telescope, right? There are mirrors, there's a focus, right? The farthest is seen, it's about 14.2 in redshift, which is a astronomy jargon of how to convert age to some unit, okay? And that corresponds to the age of the universe at about 300 million years. We want to go to at least 100 million years after T0, which is a redshift of 30. It doesn't sound like much, right? Just 200 million more years. It's not linear. It's not a linear problem, okay? We need about 100 meter large mirror, collecting area, primary mirror, to be able to see those first stars in space, okay? It's not practical. On Earth, on ground, there's an upcoming telescope called Extremely Large Telescope, ELT, in Chile. And that on Earth, with all the logistics that you can do, has a mirror that will be of 39 meters. Okay, so it's more or less impossible to actually image those very first stars and galaxies. So we use something else instead. I just told you that the universe is basically filled with hydrogen and helium. Hydrogen is very, very abundant. Okay, it's almost 92% of the gas in the universe is hydrogen. 8% is about helium. Everything else is barely nothing. Hydrogen, neutral hydrogen, has a very nice property. It's well studied. So it's a very simple system as far as me, as somebody who doesn't study hydrogen is concerned. People who study hydrogen may disagree. It has a proton and an electron. The proton and electron can be positively aligned in spin or negatively, like anti-aligned, okay? So the anti-aligned state is a more favorable state. So naturally, hydrogen with its own Einstein's coefficient, it has a time constant, will naturally go from a aligned to anti-aligned state if left alone. This is a highly unfavorable process, but it happens every some n million years. But the universe is 13.8 billion years, there is enough hydrogen, we have enough amount of this aligned to anti-aligned happening to be able to use hydrogen as a way to study the universe. And this is routinely done. We study our galaxy with hydrogen, we study other galaxies using hydrogen, using this signal. This signal, so when I say signal, this aligned to anti-aligned process, because there is an energy difference in the state, it emits a photon. That photon at rest frame has a wavelength of 21 centimeter or a frequency of 1420 megahertz, 1.4 gigahertz, okay? So we want to use the 1.4 gigahertz emission from neutral hydrogen coming to us all the way from February, basically when the first stars formed, trace that process all the way to maybe March, April, okay, and see what's happening with all the hydrogen in the universe, and that will tell us the process of formation of the first stars and their evolution over the first generation of stars, and that's what we do. So we no longer are talking about optical light and optical telescopes and mirrors. We are now talking about radio telescopes, okay, operating at radio frequencies. Okay, this signal from the first stars, the 21 centimeter red-shifted hydrogen from the early universe, from the first stars, has not been detected. So the title of my talk was how we can detect them and not how we have detected them. This is an unknown open problem that many groups across the world are working on and trying to detect this 21 centimeter signal from the early universe, from the first stars and galaxies. The signal is very, very weak. I am going to tell you a bit more about the problems about why it has not been detected, even though people have been trying for at least 25 years. Okay? So, some clues about the signal. We are thinking about the average evolution of hydrogen in the universe and how that changes when the first stars come into the picture. So, this signal comes to us from all directions in the sky. Some people try to do some more specialized, this direction, that direction, but for this talk, the signal we are trying to look for comes from all directions. It's not one star here, one galaxy there, no, okay? So that's an advantage. The second, the frequency range has now red shifted, okay? We are all familiar with school, high school, college, Doppler shifting, right? Doppler shifting is because of velocity. Cosmological red shifting is because the signal itself gets kind of stretched when the space-time expands, okay? There's a subtle difference in why the red shifting happens, but the formula is more or less, okay, the same. So what was emitted at 1420 megahertz gets redshifted to you and comes to you at a lower and lower frequency. So based on how far the redshifted signal comes to you, the signal can be anywhere between 10 megahertz to 200 megahertz. It's a very, very weak signal, partly because, well, it's coming from far away, okay, and partly because this is a forbidden transition. We measure power in terms of temperature in radio astronomy. It's jargon, it's a convention. So the signal that we are trying to detect, I have put some numbers here, but it kind of, we can work out the details later. The order of magnitude, the temperature equivalent of the signal amplitude is about 10 millikelvin, and over a frequency or a bandwidth of 1 megahertz, the kind of power we are trying to measure is 10 raised to minus 19 watts, okay. That's 1/10 of a billion watts, okay. That is how faint a signal we are trying to detect to see that signal. When I keep saying signal, what is it that I am trying to study, right? This is what it is. I will quickly go over what the x and y axis are, but I will tell you the takeaways, okay? And if you want to chat more about it, we can talk later. X axis is that redshifted frequency. Y axis is if I give you an equivalent temperature for the signal from neutral hydrogen, that spin flip transition, right? The 1420 megahertz signal amplitude. If I give it some equivalent temperature units, and I see that I am looking at the signal with the cosmic microwave background as a contrast. It's a differential brightness temperature, okay. And you see some interesting features, it goes up, goes down. That contains the actual details that we want to study. Each of these curves is a simulation of what that signal could be like, okay. The kinds of information that we are able to interpret from this curve or any one of these curves is when those first stars and galaxies formed, they emit Lyman alpha radiation, okay. How strongly coupled was this Lyman alpha radiation to the 21 centimeter signal? When did they start going, say, supernovae, and when did they start emitting X-ray radiation? How strongly did that X-ray radiation heat the hydrogen gas? And when did the entire neutral hydrogen in the universe become reionized? We have other ways to confirm that when you look at the universe on large cosmological scales, we no longer have much neutral hydrogen left. Excuse me. It's mostly ionized hydrogen, okay. So this curve will tell us all of that information. Each of this has a slightly different parameter put into it, and the name of the game is you have to find which of this is the real curve, okay. And maybe there is something completely out of the box I have not put in and it's a different signal, okay. So this is how clueless we are about that process, and that's why it's a very interesting topic to work on. So other takeaway is frequency range. I told you. I have zoomed in on the part that goes from 55 to 200 megahertz. There is another signal also at lower frequencies. And the y-axis order of magnitude, it's about 10 to 100 millikelvin. It's negative because I am looking at it in absorption, okay. I am doing some A minus B, and if B is greater than A, I will get a negative value. Now, this is an analogy that I find helpful. You drop something in a water pond or something, it creates some ripples, waves are going away, so it creates a faint disturbance. Imagine each of these drop, the thing that you are putting in is like a star, the things that are moving away, that you are trying to study the object that fell into the water pond by looking at the ripple. So we are trying to study the first stars by looking at the hydrogen, and it's a very small faint ripple. this is what our galaxy does. You know, this is like an ocean, huge tidal waves, you know, that's how noisy our own radio emission from our galaxy is. Okay? So, I basically said what I wanted to say, so I won't repeat it. So these are all the challenges to why this signal has not yet been detected. Okay? First is a very fundamental challenge. We live inside a solar system. Solar system sits in a galaxy. Galaxy has synchrotron radiation. Okay? There are electrons that are getting accelerated in magnetic field lines, and that creates synchrotron radiation. And over this frequency range, about 100 megahertz, that's very, very bright. Our galaxy emits radio waves. Okay? And that was a discovery of kind of beginnings of radio astronomy. That's how it was discovered. Okay? That galaxy emits radio waves. Those radio waves are 6 orders of magnitude, 1,000,000 times stronger than the signal we are trying to detect from the first stars that comes to us from beyond the galaxy. On top of that, we have other problems such as our own ionosphere creates refractive effects, absorption, reflection. So at radio frequencies, it is like a disturbing medium for you. Then to top it all off, we have human beings who create loud radio noise. This frequency range, if anyone knows 94.3, 95, that's the megahertz frequency of transmission of these radio transmitters, FM radio transmitters. And there are megawatt transmitters across the world, okay, it's huge, okay. All of this creates noise. So this is an actual measurement. X-axis is frequency, y-axis is some power level. And you see all those spiky lines, that is human-made terrestrial radio frequency interference. So these are all the challenges. And just to give you a picture of what our galaxy looks like in radio, this is a image probably at some low frequency, maybe 100 megahertz or so. The way to read this is like a compressed projection of a sphere, the sphere being our galaxy. The center point of that ellipse is 0, 0. 0 latitude, 0 longitude, okay. We sit somewhere along the galactic plane to the right, and in astronomy, east and west convention are flipped. Top is north, bottom is south, right is west, left is east, okay. So we are sitting there, and of course over order of maybe 100 million years or so, we take a orbit around the galactic center, okay. Don't quote me on that time scale, but we go around the galactic center. So again on the right side, these are all those curves that we are trying to detect at the bottom. What our galaxy emits is way up there. So we have to find that ripple of the hydrogen because of the drop from the first stars, and we are looking through an ocean, okay, that is the problem. Now what do we do to actually make this measurement? We build a radio telescope. For those who have not seen it, you have dish antennas on your terrace. There are many of dish antenna type radio telescopes, huge parabolic dishes. We have a few in India. There is a telescope called GMRTs of Pune, so it's among the premier radio telescopes in the world. We don't use those kinds of telescopes for this experiment. We use a very custom telescope because the problem we're trying to see is different. Most radio telescopes will want to look at one galaxy there, one pulsar there. So they need a huge collecting area with a small antenna beam that they can point. We on the other hand, we want a large beam. We don't want any localization because our signal comes to us from everywhere, but it has to be very, 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 very sensitive. So the entire detail is in the calibration, how you take that power, convert it to the right units, remove all your instrumental effects, separate the foregrounds and find that signal. So you have an antenna with a beam, it sits on some surface. You have some analog receiver, basically RF electronics, amplifiers, filters, some noise injection, calibration. It goes to a digital receiver, FPGAs, GPUs, etc. That gets collected as data, and you look at and analyze that data. So there are many experiments, well actually there are 5 experiments across the world that are trying to do this. We are one of those 5 at RRI. We have a ground based experiment called SARAS. These are some pictures of different versions of SARAS. SARAS has gone to Ladakh, it has gone to some places near Anantapur, Gauribudnur, another Ladakh, Sharavati, recently Andaman. So this is a portable, high sensitivity calibrated radio telescope. So the way we work is we take the telescope, we spent months if not a year designing every detail of this telescope, testing it in the lab, getting it to make sure that it has the necessary accuracy and precision for our experiment, sensitivity. We pack up the entire experiment including the antenna. Antenna is this big, put it in boxes, ship it. go out as an expedition, spend 3 to 4 weeks on the field, put up tents, put up batteries, generators, solar panels, and every night we run our experiment, and after 3-4 weeks we pack up everything, come back and analyze the data for the next 6 months. Okay, we've been told that a good way to describe this mode of experiment is that we are nomadic astronomers. Okay, so later I'll come to why Saras right now floats on water. But here are some nice pictures. So this is what it involves. You have an antenna that is that extremely unconventional cone-like thing sitting on some plate floating on water. A cable comes right below that, in that yellow structure that you see below that cone. You have an analog receiver that does a lot of the important calibration that I mentioned, amplification, noise injection, calibration details. Then 2 optical fiber cables come out along the water surface to a box that sits on the land that has all the digital electronics, digitization, correlation, spectrometer, etc. Now, this is what life can look like as an astronomer should you choose to be one. It sometimes involves sitting on a boat in the middle of nowhere, trying to do some jugaad with wood. Okay, so this is our team doing an deployment in Andaman, and there is a picture. Okay, I think you can see those little yellow buoys floating along the water. That is the cable going back to the shore from the antenna. And this is how fancy the setup is to protect it from rain. You put it in a tent and you sit in the tent and work. Okay. Now, so these are some places we have been to. We have gone to Andamans, Ladakh, few more places in Karnataka, and last winter a team member from RRI led India's first winter expedition to the Himadri station in Arctic, Svalbard. He was so our team member went and did RFI survey for Saras as well as Apsara. RFI standing for radio frequency interference. So when you go select a site, you have to go to a place that is not that radio loud. Okay, it is relatively radio quiet. Not too many FM transmitters, not too many telephone signals, whatnot. Now, why does Saras float on water? Okay. Here are some problems that come practically. Okay, so I showed you the simple version of that picture. This is the practical version. This is reality, and this is why the signal has not yet been detected. Everything highlighted in pink are things that are just you have to figure out in your experiment. You better do a good job. You better write good code. You better do your data analysis well. You better take care of your antenna design, all of that. But there are 3 things that are unique to operating an experiment like this on Earth. Okay. We talked about 2. One is radio frequency interference, which is human made loud radio noise that contaminates your data. The second is ionospheric effects, which can refract galactic signals. The details of this are little, I won't go into it now, but the idea is that you are looking for a signal that comes to you from beyond the galaxy. Your galaxy is very bright though, and if you have refraction, it's like you have different parts of the galaxy coming into your data at different frequencies, and that creates confusing artifacts, and you can no longer tell which part of your data is coming from your galaxy and which part of it is coming from the cosmological signal. So that's why refraction is a problem. How much of a problem has not yet been determined. It's an open problem, but what we do is we make sure we know the total electron count, or what they call TEC, which means we have a quiet ionosphere, and on those nights we observe. We don't observe during the day, we don't observe during solar flare going off, and that's disturbed the plasma in the ionosphere. Okay. The third problem is that when you have designed an antenna to do the right thing, behave perfectly for your requirements, you do all the simulations, there are electromagnetic softwares and solvers that do this for you, and you stick that antenna on soil, you stick that antenna on water, right? You stick that on any surface, right? The properties of the antenna change, because antenna is not just a piece of metal, it is finally currents flowing on a surface that you tap out at a point as a voltage. Okay, so if you put it on a surface like a dielectric, which is earth is a dielectric, soil is a dielectric, based on the things like how wet that soil is or how stratified the soil is below the top layer, your antenna properties will change, and that is a problem that is close to impossible to model accurately and extremely challenging to measure in the field. Okay. But a good way is to at least go to a low stratification medium like water. Okay. So, unlike soil whose properties can keep changing, you go to a stable water body like a lake with a known salinity, you can design an antenna for that and stick it on there and take measurements, which is why Saras has gone to water. But a better place is to go to the far side of the moon. Okay? There have been measurements by experiments in the past at lower frequencies that tell us that the far side of the moon, the side that you do not see because of tidal locking, is radio quiet. So the moon is basically a blocker of terrestrial radio emission. Okay? So you can go to the far side of the moon and you can go in orbit. So you address all the three problems that we worried about. So first of all, there is no RFI because there have been measurements and simulations that tell you there is no RFI. Secondly, there is no ionosphere per se on the moon. And thirdly, you can operate in free space. So you are not sitting on the lunar surface or soil. You are not in the vicinity of some other medium that can distort or change your antenna properties. Okay? So keeping this in mind, our group has proposed an experiment called Pratush to ISRO as part of announcement of opportunity. And we were successfully awarded a seed fund, like a grant, to develop a concept model of Pratush. So this design was arrived at after we got that seed funding. So this is a summary of what the experiment will look like. We are focusing on 55 to 110 megahertz frequency range. That conical grey structure and the funny thing below it, the umbrella below it, is the antenna. But we have to do this antenna design keeping in mind the satellite itself, because then we don't want to repeat the same problem. I design an entire antenna and stick it on a piece of metal and say work the way I expect you to, right? So we have a CubeSat dimension bus we have assumed, and keeping this bus in mind, we have designed this antenna to have all the right properties. For us, the right properties are things like having a large beam that doesn't shift too much as a function of frequency, and the way the antenna connects to its electronics is known, basically the antenna's response function, or for those who know the language, return loss, is smooth as a function of frequency. All of this is to make sure that you are able to eventually separate your galactic emission from the cosmological signal. That's what determines the antenna design. It has all those properties in the presence of this bus. Okay? Inside the bus you have the spacecraft electronics, payload electronics, digital electronics, analog, etc. There are some things, open problems that we are actively working on about Pratush. First is choosing the right orbit. We have some clues. We want to maximize time on the lunar far side without it seeing the sun and the earth, but it has to keep coming back towards the earth to downlink the signal to us. Okay? Second, we have to figure out a way for this antenna's metal to not warp, because this antenna is going to see thermal swings of 500 degrees. It will go in and out of the sun, right? And there is no atmosphere to moderate the temperature swing for you. So it's a material science problem we have to look at, right? What type of material with low thermal coefficient of expansion, right? And the third is an EMI shielding. So your own electronics are also going to create noise. Okay? You don't want to go all the way to the far side of the moon only to be seeing your own emissions. You want to look at the galaxy, you want to look at the signal you are trying to find. So the way you protect and design your electronics so that your antenna doesn't pick up EMI at the levels you care about is another problem we are working on. That is also a combination of electronics, EMI modeling, as well as material science. Okay? So what we have done is, instead of saying, okay fine, clearly we know there are not enough launch opportunities for this problem, whereas there is an active Chandrayaan program and we have been trying to get our spot in there, we can try to see if we can do this around Earth to some extent, because 2 of the 3 problems get solved, right? You can go above the ionosphere, you can go to free space, but you will see RFI, you will see terrestrial emission. But how bad is the RFI? Are there pockets of RFI that are RFI free? We are trying to see. So this is what Pratush is like in the lab now. So right now we are powering it with batteries. Eventually there will be some solar panel equivalent. All the electronics can fit on this table and the antenna is this big. The antenna size is determined by the frequency range of operation. I cannot, for the sake of flying it to space, make it a smaller antenna. It will not be an antenna at this frequency, right? So, excuse me. So this is a kind of estimate we have done. All the signals in blue, right, are signals we think we can detect in Earth orbit if all the other conditions are right. This combination of blue and red, so most of the signal space is something we will be sensitive to on the lunar far side. So this is why we are taking like a more programmatic approach and say, let's try it first around Earth, at least we'll get more launch opportunities. It is a less complex and less expensive compared to a lunar launch, and then eventually go to the moon. We are not the only ones who are trying to do the lunar far side experiment. There are every space agency, NASA, ESA, JAXA, the Chinese guys, all of us have at least 2 or 3 such proposals to try to go to the far side of the moon for radio astronomy, because we know that is where things are radio quiet. There are very few radio quiet locations left on Earth for very low sensitivity. And interestingly, below 10 megahertz, you don't see radio emission anyway, because your ionosphere reflects and blocks all radio emission below 10 megahertz. So if you really want to study anything about the universe and the galaxy below 10 megahertz, you have to go to space, which means you better go to a radio quiet location in space. So there are many competing proposals across space agencies, and it is a very tight race. So in that spirit of simulating the RFI around Earth, this is other kind of work astronomers do, well, if you can call it that. So we have models that simulate what is the terrestrial radio frequency interference that come from FM stations. So we built a global model putting in every FM transmitter across the Earth, and wherever there are gaps, we fill it using statistical models, and we think there is a clean patch kind of around the Antarctic where we may be able to get some decent radio noise free time, okay, RFI free time for such an experiment. So we want to go and actually measure this radio free part. So we have someone who is building a small payload. Right now that's just a concept, but really small, like a CubeSat type of payload that just will go and measure the RFI around Earth. This is not going to have the sensitivity to detect the final signal, but it will go and tell us what are the power levels of RFI across different points of Earth from space, and that will inform whether we can actually confidently go ahead with a low Earth orbiter full sensitivity experiment, or the only way is to go to the far side of the moon. So that's the last slide of my talk. This is the summary. I hope you got a flavor for the fact that if you really want to study the first stars and galaxies, you have to use hydrogen. You can't really be building an imaging type of a telescope. We do this at radio frequencies where there are lots of other problems. So a lot of the problem is in designing a really good high sensitivity, well calibrated single radio telescope to look and give you all the information you need to know. And this is important because that will give us a complete picture of our current universe. And also, like I said, eventually you have come from those first stars too in some sense. So it's a nice problem to think about. So I'll take questions. Thank you.
So in the sketch that you showed,
Yeah.
what do you call the area, the satellite?
Pratush, yeah. This.
Saras,
yes, yes.
So it was very small thing.
Yes.
But in Kodaikanal observatory, the telescope is very big.
Yes.
So what's the difference?
So first of all, Kodaikanal observatory is a radio observatory, so it has different design metrics. But I can tell you, compared to generally a radio telescope, we don't want a traditional radio telescope because the kind of signal we are trying to find is not a traditional signal. So we are not trying to look at one direction of the sky with a large dish, but we want, more importantly, like I said, the antenna beam of that telescope should be very stable over the full frequency range, and it should be like a badly behaved antenna across the entire frequency range. It's not a resonant antenna, because resonance introduces funny shapes in your data that you can no longer use for analysis. So it's a intentionally unconventional telescope, and we do what we call as absolute calibration. So most radio telescopes look at a different radio source in the sky and derive the calibration. We calibrate our telescope to go from whatever digital units to a temperature value, and that is a priority for us because we want to get the temperature. So that's the main difference between this type of telescope and the other radio telescope.
And one more question. So you mentioned that new stars are born and the sun is also burning.
Yes.
So suppose sun burns up.
Yeah.
Will there be any other star coming up that can sustain life on it?
Well, we will be long gone by then because the sun will grow into an envelope that will absorb the earth. So earth will be gone, or habitability of the earth will be gone when the sun dies. But we don't think new stars are born in the same location where the old stars are born. So even in the local universe, the star formation is an active research area. to see what makes a star and not a Jupiter. Jupiter came very close to being a star, so, but we don't think a new star will replace the location of the sun. It will happen somewhere.
Any more questions?
I think there's one at the back.
So ma'am, as you said that finding about the first stars is like finding the source of the ripple that is spread through an ocean.
Yeah.
So the ripple is basically a 2D acoustic wave, the ripple.
Okay.
And the droplet would be the source injection. So there's been work like using physics-informed neural networks called the inverse problem, where we have the data, observe values of the ripple, which is like at the receiver, and we measure the pressure, and we estimate where the source location was. So looking at this analogy, what data are we looking for in the form of as we are saying the ripple is a 2D acoustic wave. So what data are we looking for exactly apart from the radio waves for the estimation of the first stars?
Okay. So first of all, let me clarify that that was a very bad analogy because what we are looking for specifically in this case is not an acoustic wave. Okay, we are looking for standard electromagnetic TEM, transverse electromagnetic radio waves. That was just an example to tell you that we are using hydrogen instead of directly looking at the stars. Okay, and just to show you the order of magnitude of the noise. Okay, coming to the what other probes, what else can we use if that's the question. For the very first stars, there's honestly nothing much else we can use other than hydrogen. For the later stars, the part where the first stars start dying, right? Okay. So maybe from the point where the graph starts, let's look only at the black curve. From the point where it starts to where it comes down, there is nothing else you can use other than hydrogen, this signal. Okay. From the part where it starts going up, right? You can use telescopes like James Webb. It goes close enough to that point. You can also start looking at X-ray radiation because these are X-ray sources heating the gas. There's there's something called the Lyman alpha forest, which is basically looking at the Lyman alpha signal, but you need the first star to have already formed for that. Okay, you can look at the Lyman alpha emission coming to you through pockets of neutral hydrogen to map something about that star. But the very first stars, that period, you can't use anything else. But please don't keep in mind the analogy that it is an actual acoustic wave. It is a TEM wave that is just to get you a feel for how problematic the detection is. Yeah. I think one more question. One more here. Yeah.
I have two questions.
Yeah.
So initially my first question is, in Pratyush experiment you were saying that you needed a material with low coefficient of thermal expansion.
Yes.
So what was the material you used and how was the fabrication done?
So right now we only have the laboratory model, we don't have the flight model. So that is an open problem we're working on. So in the lab we just use aluminium. We use aluminium for all our telescopes. We have been told we can consider carbon fiber reinforced plastic with strips of metal because you do need a conductor. But I believe CFRP is a material people have looked at which is reasonable at doing this because this is not the only experiment that has done orbiting. But our constraints on the amount of distortion, warping we can tolerate is very, very low compared to other telescopes where other just communication antennas. Our requirements are very stringent. So we are looking at this a lot more seriously, but that's a possibility, CFRP. Yeah. One more at the back.
Thank you, ma'am. In Ceres and other parts, the parabolic antennas or horn type antennas are there.
Yeah.
I hope.
Yeah.
And why don't you try for microstrip antennas?
This frequency is very low for a microstrip antenna. So typically microstrip used at few gigahertz. A microstrip, people have been looking at it, but yeah, it will be like this big microstrip first of all because it's a meter wavelength frequency range. It will be huge microstrip. And secondly, we worry about some losses is one problem. And the third main problem is like I said, our requirements of the antenna is this beam stability as a function of frequency and return loss values. So we have used microstrip antenna at a higher frequency experiment with the same requirements. It is just more practical. The antenna operates at 2 to 4 gigahertz, the antenna is like this big. But it's more a problem of fabrication of a PCB this big, more or less. Yeah.
Thank you.
So I'm unsure as to how quickly the temperature change happens for the antenna.
Oh yeah. Okay.
What if, since the window for your temperature change and your distortions is very small, how about you use two antennas, one for the cold side when you are inside, when you're on the dark side of the moon, and one for when you are facing the sun?
See, partly it's because you're going to be orbiting. Right. You cannot, if you have to orbit to get your data out also, right? There are some telescopes that are planned as landers that will always sit without seeing so much of a thermal shock. But remember, the moon has a 14-day period. So every 14 days, sun will rise on the moon. So at some point, the sun always comes to the far side. The far side is not a dark side like it's popularly thought. At some point, after 14-day cycle, sun will come even on the far side. Everything will get hot. So there is no escaping that heating cooling, even if you put two, because things go round. So that's the problem.
If there are no more questions, so let us thank